Okay. Morning, everyone. Wow. It's great. Okay. So my name is Guilherme. Morning. It's my great honor to introduce Professor Grant Jensen for this early session. So I have a few words about him. So Professor Jensen studied physics and math at Brigham Young University. And he entered the MD PhD program at Stanford and earned his doctorate in biophysics, working on electron microscopy of RNA polymerase and other complexes with Dr. Roger Kornberg, a Nobel laureate for the structural studies of transcription. And he continued his works in electron microscopy at the Lawrence Berkeley National Lab and became an assistant professor of biology at Caltech in July 2002. Since 2010, he's a full professor and is now back to Brigham Young University, right? And his interests include electron tomography of whole cells and structural biology of the HAV and the development of cryo-EM technologies. Jensen was awarded Serious Scholar in 2004. He's the chair of the American Society of Microbiology in 2007 and a Howard Hughes Medical Institute investigator in 2008. He has published over 200 papers in hot topic journals, and his lab has developed a searchable tomography database containing approximately 50,000 cryotomogram and over 200 different viral and microbial samples. And it's also worth mentioning that Grant is very active in teaching with his famous course, Getting Started with Cryo-EM, and it's all available on YouTube, right? And that has inspired thousands of students and cryo-EM professionals all over the world. So with that, on behalf of the entire scientific community of our university, it's my pleasure to welcome Professor Grant on his lecture, Electron Cryotomography, Progress and Potentials. So welcome, Jen.
Well, thank you, Guy, and good morning to everybody. I'd like to thank the organizers for inviting me. I've never been to Brazil before, so I was really excited to get the invitation. And congratulations to the whole community on acquisition of such a great microscope and all of the things that you've done and will do now in cryo-EM. That's a really exciting to be part of it. I had one student in my time at Caltech from Brazil. His name was Davi Ortega, one of the smartest people I've ever met. So I always wanted to come to Brazil and see where all the smart people came from. So I'm going to speak about electron cryotomography. I want to begin by saying that I decided to join this field in about 1999 or 2000 when I was a postdoc in Berkeley, and Professor Wolfgang Baumeister came and showed tomograms of a cell. And immediately in that seminar, I knew that is what I was going to do. So then I went back to the lab. Ken Downing was my advisor, and I said, you know, I'm dropping all my projects. This is what I'm going to do. And I need to go to Germany. And so then I would go to Germany, and I learned how to do it in Wolfgang's lab at the hands of Jürgen and his colleagues. And so I just wanted to give a shout out to the tremendous group in Martinsried who allowed me to learn how to do this. And then I got started at Caltech. So these were very early days, and I thought, well, what should we look at? And I said, well, how about E. coli? It turns out at the moment, in those years, if you searched how many papers there were about E. coli, it was 10,000. It was the most popular cell to study. Turns out it was a little big for tomography without a fib mill at the time. So I said, well, what's the second most studied cell? B. subtilis. It's even worse. There's 1,000 papers, there's 10,000 papers. No, there's 1,000 papers about B. subtilis, but it's even bigger than E. coli. So what's number 3? It turned out it was Caulobacter crescentus. And that had 100 papers, so it's like an order of magnitude lower. Anyway, so we got Caulobacter, put it in the microscope, took our first few tomograms, and there was like 4 or 5 papers of discoveries, right there in Caulobacter that people had never seen. And we just went from there. It just one project after another, getting a chance to look inside cells like no one's been able to do before. Just tons of fun along the way. So what I wanted to show today is a couple basically biology stories that are very recent, including one that's not published yet, that illustrate the progress and the potential of cryotomography. And since this is the last day of the conference and I'm one of the last speakers on this subject, I thought I'd use the second half to talk about some of the technical barriers that we face now and opportunities to overcome them, and just a little bit of conversation about where we're going with this field. I hope that works well. So one of the cells that we looked at eventually was Legionella pneumophila. It causes Legionnaire's disease in humans. And it turns out if you take tomograms of these cells, at the pole of the cell, there's a very interesting and amazing molecular machine called the type 4 secretion system. So these are slices out of 3 tomograms where you can see cross-sections of this type 4 secretion system that sits between the outer membrane and the inner membrane. This secretion system is a part of a superfamily that does conjugation in bacteria. There's one bacteria that can squirt DNA into a plant cell using this machine. Many pathogens use it to squirt out effector molecules to affect the biology of the host cell. And of course, it's critical in this Legionnaire's disease, so it's of high interest. And Debnat Gosal, when he was in my lab, produced the first subtomogram average of this machine. So here it is. This is the outer membrane on top. This is the inner membrane down here. And these are the densities that are consistently present. And this isn't the highest resolution subtomogram average ever done, and this is actually a tough one. Legionella is rather thick. We could do better if we fib milled them all, but as you'll see, I think you'll see that we were able to address many questions even by looking at cells in their intact state. Also, it's a flexible machine. And so one of the things we had to do is we do a local refinement of just the cytoplasmic densities, and then separately the periplasmic densities, and then put them together. That's why we have our dotted yellow line there. So it's a challenging biological target. Anyway, in 2017, we had our first subtomogram average. And then we tried to dissect it, as we have done for some other machines, by looking at constructs where one of the proteins was missing or a different protein was missing, or cases where we had a protein linked to the superfolder GFP, and we looked for the extra density. And I won't walk you through all that, but we did lots of other structures, and this led us to an architectural model of where the different proteins were in this machine, which turned out to be largely right, but not completely right. At the same time we were doing that, our colleague Jun Liu, his group at Yale, was doing similar work addressing the proteins in the cytoplasm. There's 3 ATPases that work there, and they were able to dissect and figure out where each one was. Beautiful work that they published in 2018, showing the location of proteins in the cytoplasm. Then in 2020 and 2021, Melanie Oi's group was able to solve single particle reconstructions of subcomplexes within this larger macromolecular machine, and they reached resolutions where they could fit models. And so that was a really helpful addition. What we did next is we discovered when we were looking at these different mutants that this particular mutant, where the protein .F was fused with the superfolder GFP, it had the most stable structure. You can see more details here than in the wild type. And so we decided to take more images of this mutant, and in fact take thousands. So we recorded thousands of tomograms, which resulted in about 6,000 particles of this particular fusion construct. And so then having 6,000 allowed us to classify, and this turned out to be critical. 72% of the particles looked like this, where the periplasmic densities were fairly well resolved, but the cytoplasmic densities were not. We had another class, about 14%, where the cytoplasmic densities looked like they were missing. And then the final 14%, the cytoplasmic densities were quite clear, and so were the periplasmic densities. So our interpretation is that these classes contain a bunch of different kinds of particles that are not fully assembled or not rigid, and this class is majority particles that are more completely assembled or more structurally stable. And so we proceeded to move forward with just these 14%. These are cross-sections through the cytoplasmic densities. So moving forward with just those 14% of the particles, we further did local refinements. The first refinement was just on the paraplasmic densities to improve that average. The next refinement was on just the regions that were crossing the inner membrane, and the third region was the paraplasmic densities themselves. And this corrected for basically flexibility in the particles so that we could get the most data out of each region. And then we combined them into a composite map that maybe very few particles actually look exactly like this because they're flexible, but nevertheless, it's our best representation of each region. So here it is in the end. The outer membrane aligned very well, and so we can see the two leaflets of that bilayer. The inner membrane was not always in exactly the same position, so we did not resolve the two leaflets of the bilayer. And then here are the other densities. Now, that year, this was now 2022, just last year, Gabriel Waxman's lab shared just an amazing, fantastic result, which is a single particle reconstruction of a relative of this family of machines. We call it the conjugative type 4 secretion system. So it's part of this type 4 secretion system family, but it has a different job. What it does is it builds a pilus, and then it's thought to pump single-stranded DNA through the pilus into a target cell. Now, I'm actually not convinced that it goes through the pilus, but that's an entirely different story. I just want you to recognize that this type 4 secretion system shares many of the same proteins and homologs, but at its core, it's quite different because this one builds a pilus and pumps DNA through. Ours in Legionella is not thought to build a pilus. We have no reason to believe that it ever does build a pilus, and instead it pumps 300 different protein substrates into the target cell. But nevertheless, okay, so that was a big advance. The other big advance that happened just as we were working on this is people started trusting AlphaFold predictions, which was helpful. So we had these 7 proteins whose structures were not known, and we were able to use AlphaFold to get good predictions of their structures. Now, you know that AlphaFold will also give you a confidence score for each region of the protein. So we have very high confidence, that's an alpha helix. The rest of it is lower confidence. You know, there's a high confidence area, the rest is lower confidence. So that's also helpful to know which parts are almost for sure right and which ones are still uncertain. Anyway, with these clues, we were able to build what we call an integrative atomic model of the whole system. And this movie just gives you a flavor for what that model looks like and the confidence level that we have. So this is Melanie Oi's single particle reconstruction of the Legionella system. And the degree to which our subtomogram average envelope matches the atomic model gives you a sense for how reliable it is. It's not perfect, but nevertheless, you can have high confidence, for instance, that those proteins are in the right place. And so the single particle reconstruction of this outer ring docked very well. It's a rigid piece of material, and so that was easy to find. Now, I told you that we had fused superfolder GFP to the protein .F, and it turns out there's 2 copies of .F there, and it's a 13-fold symmetric. So in total, we have 26 more GFPs present in our model, and sure enough, we have a density right there that makes sense that we interpret as those GFPs. This we've called the plug before, and we were able to show that the AlphaFold model for ICMX, and we had a deletion mutant that told us ICMX was there, and it turns out the AlphaFold model, if you build a pentamer, fits very well into that. The main channel part we discovered is this protein called ICMF, and it's a structural homologue of a protein TSSM, which comes from the type 6 secretion system, and I'll say more about that. But that was quite a surprise to discover that ICMX forms the channel. Now, the AlphaFold model needed to be adjusted because we know this region is a transmembrane domain, and that puts the last domain in the cytoplasm here, and we discovered a new density that had not been seen before where that docks reasonably well. It binds in homologous structures to a homolog of a protein .u right here, and so that allowed us also to localize .u in the system. Now we'll move into the transmembrane region and the ATPase complex. These proteins have strong homology to proteins in Gabriel Waxman's conjugative type 4 secretion system structure. So these three proteins are clear matches. So we were able to take those atomic models and build by homology there. This is our protein in the Legionella type 4 secretion system, and this is the one in Gabriel's structure. And so we match the regions that do match. There's some differences, but there's a lot of similarities. And using that, June's lab showed that this protein forms a hexamer of dimers. And so we build the hexamer of dimers to fill in that map. The cytoplasmic densities are probably the most flexible, and so there's the largest mismatches between the cryo-ET envelope and the atomic models. Anyway, we see that there's a linker with some transmembrane domains, and then this part is in the periplasm. This turns out to be a complex of two .i's and one protein called .j. It mimics something that was seen in the conjugative type 4 secretion system, so we have high confidence of that, and that leaves this dimer of .I to fill these domains in the periplasm that we see. So we're almost done. The last protein at the bottom is a clear homolog of something in the conjugative system. It's .B. It's also an ATPase. Fits very nicely into that density. June's lab first showed that that's where it is in the system. And so together this allows us to build an atomic model of this enormous machine. It's an integrative model. Obviously some of the domains might rotate a little bit, but we know where they are, which ones touch which ones, and allows us to make hypotheses about how it works. So in terms of numbers of atoms, you know, it's bigger than a ribosome, but smaller than a nuclear pore complex, if you just wondered how this fits into the world of huge structures. Bigger than a ribosome, smaller than the nuclear pore complex. So here it is. So what did we learn about the structure and the function? Well, let me remind you that, so this is Gabriel Waxman's structure of the type 4 secretion system that builds a pilus and pumps DNA. Here's our structure of the Legionella type 4 secretion system, which does not build a pilus, and it pumps out many different protein effectors. And I've color-coded homologous pieces. You can see that there are many homologous pieces. The Waxman group proposed that pillin subunits in the inner membrane would slide up the shoulder of this platform protein in gray and would come beneath this thing, which would become the tip of the pilus. In our system, there is no platform protein. There's nothing with homology to that. Instead, we have this completely new protein that has homology to a type 6 secretion system. Completely different secretion system. And this thing that used to be at the tip of the pilus seems to have moved up here and become some kind of a plug. And there's evidence that in infection, this comes out. And so you can imagine the plug being unplugged so things could pass through. Many other things are similar between the two systems, but this is the big difference, the presence or absence of the channel. So, as I mentioned, this protein has homology to TSSM, which forms the channel in a type 6 secretion system. This is the core complex of a type 6 secretion system. So these domains have homology to these domains, and I've colored them similarly. So why is that interesting? Well, that gives me an excuse to talk about something I haven't talked about in about a dozen years, but I know some of you study it, the type 6 secretion system. What a fun subject. We actually were the ones that first figured out what is the structure and function of a type 6 secretion system. Just by dumb luck. We were looking at Vibrio cholera cells because they actually have two circular chromosomes, and we thought we were trying to see how do bacteria segregate chromosomes in some kind of mitosis-like, process. And instead, when we looked at Vibrio cells, we saw these beautiful filaments, very long, and they had two different states. They were long and skinny and full of something, and then they had another state where they were shorter and fatter and empty looking. And we were collaborating with John Michelanos at Harvard, and we started to guess that maybe what we were seeing was what he had been studying genetically, this type 6 secretion system. We said, well, please put a GFP tag on the most abundant protein in that type 6 secretion system, and let's see what happens if we take light microscope pictures of it. And it was beautiful. It turns out this most abundant protein forms these elongated structures that within one frame of the movie collapse into something about half as long, and then they dissolve. And we had other clues. It turns out one of the proteins had homology to this contractile phage tail. This is a movie from Michael Rossman's group about contractile tails of phage. And once the phage sits down on its target cell and all the receptors engage, then this outer sheath undergoes a conformational change that drives the inner rod to poke through the cell and eventually get its DNA in through that way. And this led us to the mechanistic hypothesis that the type 6 secretion system is basically a javelin thrower. It can throw a javelin into its neighbor. And just for fun, I just want to show this movie because it's so fun. This is something that John's group did afterwards. They showed that if you put Vibrio cholera cells in a co-culture with Pseudomonas aeruginosa, the Vibrio cells build type 6 secretion systems in red, and they fire at the Pseudomonas cells. They don't kill them. The Pseudomonas cells sense that. They start building their own type 6 secretion systems. They fire back, and that's lethal. They'll pop all the Vibrios. So that's super fun. Thank you. In other cases, we saw organisms with whole clusters of type 6 secretion systems. So here's one with an armada of many type 6 secretion systems all loaded at the same position, probably to carry a greater payload. In one species, we had a fun result. This is called Pseudo-Alteromonas luteoviolaceae. You probably never heard of it. You probably won't hear of it again. But anyway, it does one cool thing with its type 6 secretion system. These cells, once they reach a quorum, if you put a fluorescent tag on their type 6 secretion system, what you see is that after they reach a quorum, about 1 in 100 cells seems to form a little ball of type 6 secretion systems. It turns out those cells self-destruct, lyse themselves, and they release this ball. And what is that ball? One of the most fun things we ever did was to take tomograms of those purified clusters. And it turns out to be about 100 type 6 secretion systems all linked together. They're a lot like phage tails, but they use their tail fibers instead to link together into a hexagonal lattice. We called this the Death Star. So what these bacteria do is 1 in 100 just converts their entire contents into a Death Star, and then they lyse themselves to release it out into the environment. And how about that? What's that, Chuck? Why do they do this? I don't know. What's known about this Pseudo-Alteromonas luteoviolaceae is that the reason anybody even cares is ships in the ocean, they get fouled. You know, barnacles and all kinds of stuff grow on the side of the ship, and then the Navy has to scrape them off so the ship will go quickly through the water. And this has been discovered that one of the things that sticks to the ships and grows on it uses these Death Stars as a signal that this is a good place to sit down and live, because there's a lot of bacteria that are thriving here. So that's not why that's not the selective advantage why they evolved this thing, but we can guess, any of us can guess. Anyway, that's the type 6 secretion system. I know some of you study it, it's fun. But so what does this channel do? Well, its purpose is to open up and eventually allow the entire javelin, that inner rod, we had a talk about that earlier, to pass through. It's a channel for large cargo. with a big conformational flexibility. And so it's no surprise then that we see in the core of the Legionella type 4 secretion system that this is a hybrid machine. A lot of pieces of a type 4 secretion system, one core piece of a type 6 secretion system to become a channel instead of a pilus platform, a pilus building platform. And we presume that substrates are pumped through the ATPases through this channel, the plug gets ejected, and that's how things get out. There's a lot left to be learned. So I told you the story because it illustrates some of the progress and potential of cryo-ET. First of all, we've worked hard to try to increase the speed of getting data for cryotomography, and that was important because we needed thousands and thousands of particles to be able to classify them. And so we are so grateful for the development of powerful software that allows us to classify the particles correctly. It also illustrates how you can use local refinement to do better on a big flexible machine and get each part of it as well resolved as possible. It illustrates that the world is different now that we have AlphaFold. I think the combination of AlphaFold and cryo-ET is going to be a ton of fun for the next couple decades. We're in a good position to have fun. It also illustrates the power of single particle cryo-EM. Since the resolution revolution, now we're getting more and more pieces of these machines by single particle reconstruction, and that's essential. In the end, it's one of the largest structures that has been built. It's an integrative model. Let's remember that rather than experimentally determined to atomic resolution. It's interesting because it shows it's a hybrid machine between two large secretion systems. We've learned a little bit more about its function as I described, and then the last thing I'll just mention is there's all kinds of interesting things about signaling that need to be tested now. For instance, if all this thing was a pipe to transmit proteins into the target cell, it would not look like this. Pipes look like beta sheets, beta barrels all the way up the periplasm. That's what evolution does when it needs a pipe. This is way more complicated, so there's all kinds of fun stuff going on here. There's probably signals coming from the top when it engages a host cell that are signaled down to the ATPases to say that it's time to secrete. Or maybe it's the other way around, that when the ATPases get the signal to start secreting, maybe that causes this thing to pop open like a javelin and poke a hole in the host cell. I don't know, but it's way more complicated than just being a pipe. So at that point, let me just acknowledge the people who did all this work. Shemek Dudka and Stefano Maggi did most of the work and the modeling with help from UC. And Debnat got us started with Legionella and did the first reconstructions. Phyllis Wang is a graduate student who continues to work on this. Martin Pilhofer was our lead researcher on all the work on the type 6 secretion system. Some of you know Martin now. He works at ETH. And Joe Vogel was our important collaborator on the biology of type 4 secretion systems in Legionella. So that was a story that illustrates kind of the forefront of what you can do with subtomogram averaging of a complex, large, flexible machine and AlphaFold and single particle structures. Now I want to show another result just because it's fun and it illustrates the boundaries of cryo-ET. This one is in human cells, and it's about a protein that senses protein folding stress. The protein is called IRE1 alpha. And this is a movie from Peter Walter's lab at UCSF. It's a human cell, and there's a fluorescent tag on this protein IRE1 alpha. It's in the endoplasmic reticulum. And what Peter's lab discovered is that if you add a drug that causes protein folding stress, IRE1 alpha co-localizes, it starts to aggregate into distinct puncta. And then if you wash the drug out, eventually it diffuses back throughout all of the ER, and you no longer see puncta, you just see labeling the ER across the cell. And when he saw the kind of work we were doing, he asked to collaborate to see if we could help him get pictures of what those puncta look like. And it turns out it was quite rewarding. So I'm going to show you a movie that shows CLEM results This is a big human cell growing on an EM grid. Of course, you recognize the quantifoil holes. The cell is expressing green fluorescent protein tagged IRE1 alpha. And so the cell grew on the grid. We treated it with the drugs that cause protein folding stress. IRE1 alpha aggregated into its puncta, and then we froze it. We also added these blue fluorescent beads, fiducial. I think it just, okay, you got it back on. So that's what you're looking at, a plunge frozen cell, and this is the cryofluorescence image. Okay, now we're ready to roll. This, by the way, is the nucleus. That's why there's not much IRE1 alpha there. And so with the cryofluorescence image, we get such a picture, and now we zoom in with a EM image, and now I'm showing the tomogram of that area with the fluorescent superimposed, so you know this is the place where the IRE1 alpha puncta were. So what do we see? I know that went by a little fast. I'm going to back it up a little bit. So what you see where the fluorescence is these very thin tubes with what looks like a railroad track going through them. And these tubes have three-way junctions, and they emerge from the ER lumen. So let me give you a little bit of orientation. These areas that don't have much material in them, that's the lumen of the ER. The areas that are full of more material, especially ribosomes, that's the cytoplasm. And these tubes come off of the ER lumen, and they look like they have little railroad tracks in them. So it turns out if we focus on just one of those tubes, then we get the following. And that goes too fast. If we go back through that, what we see in these tubes, let me orient you again. This wider material, this is the lumen of the ER. Then here's the ER membrane. This is cytoplasm. It's more densely packed and has ribosomes. And then this is a thin tube coming away from the ER. So again, this is ER lumen. And what we see is this railroad track-like filament in the middle. And in some areas, you'll see as I go up and down through it, it looks like the lines are going from 10 o'clock to 4 o'clock. And then when I hit another angle, it'll look like it's going from 7 o'clock to 1 o'clock. Let me do that now so you can decide whether you agree with me. So as we go through that, from top to bottom, you can see it's actually a helix. It's forming a helical structure. And of course, this is the tomogram of one region of a cell. This is not an average. And then these helices sometimes make 90-degree turns, and this one is turning down into the tomogram parallel to the z-axis, so is this one. So they can bend quite dramatically and turn the corner and change direction by 90 degrees. So we were able to visualize directly that it's a double helix. When we did this subtomogram average, we can get the pitch and the diameter quite well. There was a crystal structure of this protein in yeast, and it crystallized like this, but nobody knew whether that was an artifact of crystallization or whether it was biologically meaningful. Our interpretation is that in the human, anyway, the double helical structure is biologically meaningful. The human, it has a different pitch. We don't know if that's difference between human and yeast or if that's just a crystallization artifact, but this is what it looks like in the human. And the biology works like this. This is a diagram where here's a membrane, and this is the ER lumen above, and then here's the cytosol in below. And this protein, IRE1 alpha, has a luminal domain at its C-terminus, and a linker, and then a transmembrane domain that comes out of the ER, another linker, and then a kinase domain and a regulatory domain. And so we see that when there's protein folding stress, that causes the luminal domains to form this tight double helix. That's why it aggregates into a single punctum. What that would do biologically is it would concentrate a lot of the kinase domains and regulatory domains right outside that tube. And these cross-phosphorylate each other to activate. And so bringing them all together into a small spot is what activates the signal to the cell that there's protein folding stress. That project was done almost entirely by Stephen Carter when he was in my lab, and it's a collaboration with Peter Walter and Han Tran, Peter's student. Okay, the last story I'm going to tell before we get into the technology stuff is just illustrate something fun about cryotomography, which is that it can see transient structures. So this is a tomogram, a famous, in our lab, famous tomogram that was collected by Iway when he was in the group of Budello vibrio. And here's a highlight of its flagellar motor. And it's fun to see, like this domain and this domain, this is part of the C-ring. And you can see the axis of the motor is right through here. The C-ring is actually offset. So you can see something about conformational flexibility within, by visualizing an individual molecular machine. Our group and others have produced subtomogram averages of flagellar motors from many different species, just to give you context of where we are in this project. They all have the same core, but there's very different decorations in different species. And the assembly process of flagellar motors is complex. I don't want you to try to capture all of these detail. I just want you to know the assembly of the motor has been worked out over decades, and it's complicated. And what's fun about tomography is that a postdoc working with me, Mohammed Kaplan, I'll show his picture in a second, got good enough at recognizing flagellar motors in our tomograms that he started recognizing assembly intermediates. So this is the first one that he could see. He calls it the MS complex because it has the MS ring, and then it also has FLHA in the cytoplasm, and it has the first protein of the C ring called FlyG. So here's a schematic. This is a subtomogram average of several, and the schematic, that's the first one he can recognize. Then he can recognize the next step, which is when the other two proteins join the C ring. He can recognize the next step where it starts to elaborate the rod. Here's the next one where it's now been joined by what's called the P ring, which navigates the peptidoglycan layer. And here again we had enough particles to do a subtomogram average. The next stage is when it pokes a hole in the outer membrane using the L ring, and so now you can see a hole in the outer membrane. Then it builds the hook. Then it elaborates the filament, but these particles still are missing what we call a collar density. And the last stage has everything, including that collar density, and we have hundreds of particles like this. So that's the best subtomogram average. So it's really cool that you can see assembly intermediates inside the cell. Now, the reason that matters so much is that Mohammed discovered a new component to flagellar motors that no one had ever seen, because it's a chaperone that helps the thing assemble, and then once it's assembled, it goes away. So all the people that have purified flagellar motors, this was unknown that it was part of it. It's the one that's highlighted by all these blue arrows. There's a ring on the outside. Here's Mohammed's picture, and here's a subtomogram average of one of these assembly intermediates. It's this ring outside the C ring that no one's ever known exists before. And he discovered it by imaging assembly intermediates. So now we need to identify it and understand what role it plays in the assembly of this awesome machine. He was also able to identify disassembly intermediates. Turns out once a cell digests its flagellar motor, almost all cells leave a little relic in the outer membrane, the P and L rings. Here's a subtomogram average of that relic. So tomography allows you to see intermediates that could never be purified, I suppose. Maybe, should never say never. Maybe there'll be a mutant that stalls it in that state and it could be purified. Okay, I'm going to spend the last minutes on just some thoughts about the future of this technology, where the boundaries are, and ideas of how we could make progress. Obviously, we'd like to understand biology all the way from small proteins to tissues, and I think I've shown how cryo-ET is a fabulous technique to investigate the structure of small cells. But today, the most popular ways to look at large cells and tissues is light microscopy or thin section EM. For large complexes, it's single particle cryo-EM, and then there's X-ray crystallography and NMR on the small side. But what I'm arguing is that because of the development of fib milling by Jürgen Plitzko and others, I think cryo-ET is going to play a very important role in understanding large cells and tissues as well. Further, I think once we're able to collect tilt series in a matter of seconds, I think cryo-ET is going to play a large role in the analysis of purified large complexes. You know, the domain right now of single particle cryo-EM. Finally, this is a tilt series we took of a tiny little crystal. The kind of crystals, they're actually even smaller than what Tamir Gonen is looking at with micro-ED. But it's a tilt series of a tiny little crystal. We call it a nanocrystal because it's only maybe 50 nanometers across, 100 nanometers across. And here's the tomogram of that where you see the individual unit cells. I think this, there's some real technical hurdles to be overcome, but I believe that this will be a good way to understand the structure of large proteins and small proteins through tomography of tiny little crystals that are maybe terrible as crystals. They might have breaks and twinning and any number of problems with the crystal. We could probably solve those with image processing of a tomogram. So as a result, my prediction is that cryo-ET plays a large role in the entire spectrum of structural biology. Now, is that because I do cryo-ET? 10 minutes. Well, here's some fundamental arguments to think about as you go home and ponder whether I'm crazy this weekend. First of all, electrons are just better than X-rays for structural biology. This has been argued by Richard Henderson and others. First of all, the information to damage ratio is better. Also, but mainly, electrons can be focused, giving you phases. You get twice as much information with electrons than X-rays. So if you can do it with electrons, it's going to be better. Second of all, it's always better to have 3D data than 2D if you can get it. And there's this dose fractionation theorem, blah, blah, blah. Anyway, this is true. 3D data is better than 2D if you can get it. That's why 3D imaging with electrons, aka cryo-ET, is going to play a huge role on all scales. But for this to happen, many technical challenges remain, and I don't want to underplay the fact that there are major technical challenges. So I was going to just throw out a few of those challenges, some of the ideas that I think are exciting. One of them was working with infectious agents. That's the ones that we care most about. But because cryo-EM preserves things into a near native state, you have to protect yourself. And I just wanted to highlight a good idea that came out of my lab. What would happen if we froze the samples and then we used UV irradiation to inactivate them? UV radiation causes cross-links in the nucleic acid that kills an organism, but you probably wouldn't even see it in a cryo-EM image. And so if you can UV inactivate and image things, and we showed this works great. We took apoferritin and applied enough UV radiation to kill any cell or virus that was on the grid, and we solved the structure both before and after the UV irradiation, and the two structures, one's blue, one's yellow, they're identical. You can't see a difference, and so you could look at things in a near native state, but it's still disinfected with UV irradiation. Okay, the holy grail would be to label a target of interest like that IRE1 alpha with a fluorescent tag and watch it move and act with live fluorescence microscopy. Then just at the moment of interest, when it's doing the cool thing, plunge freeze the cells, locate it in X, Y, and Z by some kind of super resolution cryofluorescence microscopy, mill to uncover it, get a cryotomogram, locate it by Clem, and then do subtomogram averaging to high, potentially near atomic resolution, like Julia Muhammad has shown so beautifully as possible when you have lots of copies. Towards that end, Jürgen Plitzko and I and others in a consortium joined a company named Delmic in producing a prototype fib mill where you have the fluorescence objective positioned right underneath the sample as you fib mill. So in other versions of this, you have to move the sample from under the fib over to a fluorescence objective and take a picture, and then move it back underneath the fib. And when you do that, it could move a couple hundred nanometers up or down or left or right. And so what we really need is a fluorescence objective that where the sample doesn't move, but you can get fluorescence images while you're milling. And that's what this instrument does. It's called the Enzel. And we've got a prototype now. And so here's an example of how it works. This is a muscle cell with the Z rings labeled in green fluorescence. And you can measure fluorescence while you're milling if you want. And so here's a fluorescence movie while the milling is happening. Here's the trenches, to release strain. And here's the lamella that's being milled. It gets smaller and smaller and thinner, but you can see that you still have the fluorescence preserved even while you're milling. So this is going to be powerful to allow us to take a specific fluorescent target and mill to retain the target in the lamella. Now, that's possible with many instruments, but it's a question of success rate. So we've got this now, and we're starting to do some biology with it. One of the first things we're looking at is the microtubule organizing center. These are cells that have a red fluorescent marker on tubulin. So the microtubule organizing center has a lot of tubulin. This image is of an already milled lamella. So the fluorescence intensity is cranked way up, and so the rest of the cell looks terribly bright red, but this used to be the brightest red spot. Now we milled it, so it's only a couple hundred nanometers thick, and we still have some red fluorescence. And if we target that with tomography, sure enough, we find the microtubule organizing center. And the good news is that we can do this all day. Here's another example. Take the brightest red spot, boom, we hit another microtubule organizing center. Here's another one, bright red spot, boom, we hit it again. Here's another one. This time, you see now we're looking end on to this one. But we can now reproducibly take a fluorescent target, mill, to retain it in the lamella and get a tomogram. This will be powerful. Now, right now, it's not super resolution, and it takes lots of fluorescent markers. So what we need is an artificial 21st amino acid, an unnatural amino acid whose side chain is like Psi3 or Psi5. That's 1,000 times brighter than GFP. That would probably be good enough. And we also need super resolution optics in this machine so that you could have a single unnatural amino acid in a protein and identify where it is in tomogram, in a single copy. That's what we need. By the way, that work is Phyllis Wang, a graduate student in the lab, and Pete Dahlberg at Stanford is driving that work. There's challenges in data collection. I'm running out of time. I'm just going to skip this and say that tilted images are lower resolution than untilted images. And that's a major drawback to cryo-ET. We've got to solve that problem for cryo-ET to be useful across the spectrum, like I predicted. One of the problems could be that maybe drumhead vibrations on the grid are degrading the tilted images. You see, if the grid was vibrating in a drumhead motion and it's untilted, that would not disturb the image much because it's a projection. But if you had a tilted grid and it was vibrating that way, it would disrupt the resolution in one direction, which is in fact what's seen. And so how can we beat this? Maybe we can take pictures faster than that drumhead motion. I'm going to move on through that. Another data collection challenge is just the speed of getting tilt series. It's slow compared to single particle reconstruction. And this is a historical point of how it used to be really slow. And we've been able to increase the speed using the fast incremental single exposure methods, etc. You can learn about those if you want. Another way we need to increase the speed is by increasing getting larger fields of view. This is the thin edge of a pancreatic beta cell that we imaged, and all the yellow spots are places where we took tomograms because we wanted to see the progression of the secretion granules. And this led us to propose montage cryo-ET. It's a long story, but basically, what would happen if we took overlapping exposures in a hexagonal pattern? Most of the material is exposed once, but some of it is exposed twice. And if this is the pattern you took at 0 degrees, once you tilt the image, we studied what's the best way to shift the pattern or rotate it between angles so that the areas of your sample that take a double hit of dose in this tilted image are different than the ones in the first image. And so we study lots of ways to do this. Turns out you can get a tomogram where the average dose for each little part of the material is about 20% higher than it would have been if you'd just done a single exposure. But you can use that because it's useful. Anyway, make a long story short, it's possible to do this. You can take montage images and crop out the fringes. This is best done with fringe-free illumination, and stitch them together to form big montage images to make bigger tomograms. This is this was just about 27 images in each montage, and we put it together to get a tomogram of something that was a little over 3 microns on edge. But there's no reason we couldn't do this to cover an entire lamella. That's what we ought to be doing. You get a picture of the entire lamella this way. And the resolution should be just as good as a single shot tomogram, but there's a lot of image processing development that is needed per particle refinements in order to make that just as good as a single shot tomogram. Last things, we need to be able to work together and share results. We did a little bit in this direction. We made a database using blockchain technology. That's what the Brazilian gentleman in my lab did, Davi Ortega, and shared about 10,000 tomograms, but it's not what we need. We need a community repository that's different than the EMDB or this thing that we did, in that it allows different scientists to process the images differently and post the results. Like imagine if you know, I posted a tilt series and then you could post a different reconstruction than ours. And somebody else could do the segmentation and annotation and post it. And somebody could denoise it in a different way and analyze it. And everyone could share those results. And it would be clear from the database that the tilt series was mine, but the analysis was yours. So it becomes a conversation. That's what we need. And I think CZI is going to do it. So Jürgen mentioned sharing all of his awesome images of C. elegans with the CZI portal. And we're trying to get them to take as many of ours as they will. They're being a little reluctant to take all 50,000, but we'll see. So we need new alignment algorithms, etc., all this stuff. Finally, I just want to, we took one stab, an experiment in how do you share these tomograms with the world. We tried by making an atlas of bacterial and archaeal cell structure with my co-author, Catherine Ikonomu. It's like an atlas of the past where you have a great EM image on one page and then an extended figure legend on the opposite page, and it's just page after page of beautiful EM image. In this case, each image is a 3D tomogram, so you have one page of text, you have one tomogram, and the tomograms illustrate different things. This one is of Myxococcus xanthus, and it highlights our work on the type 4 pilus. Anyway, we have annotations to help people understand what they're looking at, because nobody knows unless they've been doing tomography for a while. There's little buttons you can ask for when we have more information about a structure or even how it works. We can link that in. So the book is flexible. You can get a lot of information or less depending on what stuff you click. Anyway, that's available for free to everybody at cellstructureatlas.org. And it may be a good way to share tomograms. We'll see if it works. And with that, this is just a reminder. Oh, yeah. Sorry. Whoa, okay. So there's the URL. And, you know, if it turns out to be successful, then we could, I'd be very happy to add many people's tomograms. We need to keep it coherent, but it's probably the way we need to share our tomograms with all the biologists who want to see them, who don't know how to use 3D mod. Okay, with that, I'll finish, and thank you for your attention.
Thanks, Grant, for this inspiring talk. So we have like 4 minutes for questions and discussions. Anyone? Can I have the microphone?
Yeah, you bet.
First of all, thank you very much for such a nice presentation and very inspirational information. I mean, it's very important. I just wanted to ask something about the technical part of your talk. What's your opinion about implementing dynamic or adaptive optics to all of this mixture of EM and light microscopy? Do you think this is going to be a good thing? Do you think it's going to be practical in the future? You know, these adaptive optics used in astrophysics for getting better resolution and phases. I've heard that it's faceplate that can be, you know, in this last slide that you showed, this kind of taking different positions and then making a composite reconstruction. It's kind of like that, but you are using adjusting the optics in each position just to get the best of each part.
I don't know enough about that to say something useful. And I couldn't even predict what the best super resolution light microscopy technique will be for this super resolution fluorescence guided fib milling, which is essential. So I'm sorry I can't comment on that, but I know that the keys to it are going to be getting a very bright fluorophore so that you could identify a single copy of a protein because it's very bright, and we've got to get super resolution in order to localize it within a tomogram and identify it. So maybe adaptive optics will be a part of that. I think it's quite different than my montage cryo-ET idea. The montage cryo-ET is a small perturbation to regular ET, but I think it's going to be the way to get pictures of the entire lamella. You know, you spend months and months and years preparing a sample, fib milling, and you get a lamella. It's a crime to only get tomograms of a few positions on the lamella when we really could get the whole thing. This is going to work. We didn't finish it. We just showed proof of principle. What needs to be done to finish it is software development that allows you to reconstruct subvolumes throughout the montage, because the problem is the sample itself warps and stretches during the tilt series. So the last tilt image is of a different object than the first one. And so the reconstruction has to adapt to that. We have to reconstruct subvolumes along the way, going back to the original images, and those will be just as good as a single-shot tomogram, but then we need to stitch them together in 3D, so we get, but it will be just as good as a single-shot tomogram, 5% less, because you actually waste 5% of the dose, but of the entire lamella. And it shouldn't take that long either, but it's a big computational challenge, so if anybody's looking for a computational challenge, I recommend it. Please solve that for us. Anyway, thanks for the question. Waverly and
Okay.
he's got it.
Could you comment about the possibility to get information about the phospholipid component or the lipid component of the membranes interact with this protein, the transmembrane proteins, with cryotomography?
Adam Frost at UCSF has been doing some of that. He's introduced a heavier atom into certain phospholipids and shown that they're predominantly in the inner leaflet rather than the outer leaflet in these thin tubes that escort protein produces. So I think he's at the forefront of this, and I think the answer is barely yes. He can probably see a difference when he introduced some heavy atoms into a particular phospholipid. Is that helpful? But otherwise, the lipid membranes, you just see a darker strip where the phospholipid head groups are, and then the hydrophobic core looks actually less dense than the cytoplasm of a bacterial cell, for instance. So you just see it as two lines. When it's aligned well, you resolve the two leaflets, but that's as much as you can do. People have done subtomogram averages of ribosomes actively translating a protein through a membrane, and they see the transmembrane components. So you can get that in a subtomogram average.
So, Grant, thank you very much for the
Yeah.
talk. First, regarding the resolution, I recommend you to talk to Adan. He has developed a method to improve resolution in a single image. I think that this will be really nice for the technique that you're applying. I don't know
Okay.
if you met him.
Yeah, I did. Yeah,
Yeah.
he showed me the
Yeah.
reference, so I'll
Yeah.
look at that. Right.
And my question is about this the folding problem. So this helix that you have inside the endoplasmic reticulum. What does that mean? You have a problem of folding of the proteins as they are being synthesized, or this is a reaction of the cell due to the drug treatment?
It's a reaction. The drug treatment blocks something in the protein folding process. I'm sorry, I don't remember exactly what. So the unfolded proteins in the ER lumen are sensed by that luminal domain, and it causes them to form that helix. The helix draws together the kinase and regulatory domains of many copies close together enough that they'll phosphorylate each other and send the signal. The signal comes from the regulatory domain and eventually goes to the nucleus. So does that help with the question? Yeah, it's unfolded proteins that interact with the luminal domain that cause it to form the helix. Yeah. The reason I show that is just because every single thing that forms a fluorescent puncta, we just need to go through the literature and make a list. And everything that's been shown to form a puncta, we need to do clem and see whether it's interesting. Some have not been interesting. Some we were all excited to look at, and they turn out to be phase-separated globules, and those are not interesting to look at. They look like nothing. But we need to know that when that's the case. This one was the exciting one because we looked at the punctum, and sure enough, there's this beautiful double helix. So that's the one I show. We've looked at a bunch. But anyway, our field, we need to just look at everything that's ever formed a punctum by fluorescence and see what it actually looks like at molecular resolution. Yeah. Yeah.
Okay, so here.
Okay.
I was about to say something that Larry tell, but I'm just going to finish the comment. So there is novel ways to overcome the diffraction limit, and it can be through numerical approximations like the convolution or these kind of things that now are really going deeper. So you don't need right now to so what I'm trying to say is that the H diffraction limit is not a limit at all. There is much more information in the images. There are several groups that have managed to overcome that, and we have a deterministic solution. So maybe that can be very helpful because it can be really quick, and you can get the computation right there and to combine with FIBSEM.
Yeah. Great. Let me just say that, right now, we need dozens of fluorescent proteins to produce a big enough signal to guide the milling. So we can look at microtubule organizing centers or the Z ring of a muscle, a Z band, sorry. But we should eventually be able to have a much, much brighter fluorophore so that you could see one copy of something. Then the precision to which you can localize it is going to define the limit of how big it has to be before we can interpret the tomogram, right? If you had a fluorescent tag and super resolution techniques and the things that you're developing at N could get you down to a box of, say, 30 nanometers by 30 nanometers on a side, and you knew your target was in there, it becomes a lot easier to try to recognize your protein by its density and shape if you're localized to a small box. If the box is 200 nanometers across, then you're not going to find small things. So sometimes people ask, how large does your protein have to be to find it in a tomogram? Right now, it's like 400 kilodaltons. But if we had precision clem with super resolution techniques like you're talking about, it could go much, much lower. The other thing I like to say, which I think is important, we should all say it a lot, is that when you think about a cell, there are some things that are in a particular place for a reason. The way they get there is they stick to something bigger. Then there's a bunch of stuff in the cell that just diffuses randomly. We don't need to know where that stuff is. You don't need to know where the glucose is. It's everywhere. And we can already model it, right? The only thing we need to know about the cell is where are the big things that are in a particular place for a reason. That's precisely what cryo-ET can show you. It's the big things. So we shouldn't get depressed like we're not going to understand cells just because most everything is too small to see by cryo-ET. Because most of that stuff we don't need to find. It's everywhere. It just diffuses. What we need to find is what are the large structures that have some purpose, and like a type 6 accretion system. No one had any idea what the thing was until cryo-ET showed it's a javelin thrower, right? That's the stuff we need to discover, and that is accessible by cryo-ET. Now I'm on my soapbox. Let's take another question or end as you see fit.
Yeah, we should move on. So with that, I'd like to thank Grant again, and we're ready for the next session.
Okay, so moving on to the next talk. So it's a slight change in the program here. So Fabio was supposed to be the chair of the session, but he has to handle the sound system, so he asked me to introduce Dr. Sashi. So it's a pleasure to be here to introduce Dr. Sashi. He is the director of the Hans Husker Center for Structural Biology and holds a professorship at the Heinrich Heine University in Düsseldorf. Professor Sashi embarked on his journey in cryomicroscopy while exploring amyloid fibers under the supervision of Markus Fendrich. And his postdoctoral tenure was marked by the significant research at the MRC Laboratory of Molecular Biology in Cambridge, where he worked alongside Richard Henderson and Roger Williams. So Professor Sasha has established a research group focused on unveiling the mechanisms of autophagy through the lens of the electron pair microscopy. His interest is in pushing the boundaries of single particle EM, especially in helical reconstruction, has culminated in developing the helical processing software package, Spring. His current research endeavors are at the forefront of the cryo-EM map interpretation by atomic models. This innovative work is pivotal in advancing our understanding of membrane biology, specifically in autophagy and extrinsic machine proteins. So, Dr. Sasha, thank you very much. The stage is yours.
Thank you very much for the kind introduction, and also thank you for the organizers in setting this up so wonderfully, and really thanks a lot for the great hospitality here. It's a pleasure to be here, and congratulations also to this great facility, and I'm sure this is a great home, because from what I've seen here, you really have some very nice biological systems that you can use to study with this high-end infrastructure, because I think that's also really an essential part to provide high-end science, right? So what I'll do now, in the first part, I will mainly talk about what the title is represented, the work on cryostem that we have been doing towards establishing, and also really, going into new types of imaging for cryo-EM. In the second part of the talk, I'll show some of our typical applications in membrane biology on the escort machinery, and how we address several different imaging modes ranging from single particle all the way to tomography to study these systems. Before I will go into the science, I'll briefly show you where we are from. So we are now based in Jülich, so I guess most of the people may mistake this sometimes as Munich, because the pronunciation is so similar, but it's actually located in quite another part of Germany on the western border, right close to the Netherlands and the Belgium side. So in 2018 we started here cryo-EM basically in the Ernst-Ruska Center. The Ernst-Ruska Center is traditionally a material science hub for electron microscopy, founded in 2004, and it became worldwide well known because it was the establishment of the first CC and CS corrected system that led to extremely high resolution, 0.5 angstrom resolution in TEM imaging at the time, and this is also what the center is still known for. And with the arrival of cryo-EM, what we are also venturing into is to explore these technologies like these double-corrected systems also for cryo-EM. And we are currently in the process of finalizing a building where these new microscopes in quite unique settings will be housed. So this is the room here of this Titan Pico microscope. It's a really high microscope because of these additional correctors there. And we are also at the Ernst-Ruska Center, a national user facility where worldwide users actually have access to. And this is our co-director, Joachim Meyer. That's also been very supportive in setting this up. And as I'll show you now in the next few slides, really this is also now really a nice collaboration between material science and life science EM. This is images of our supercomputer that we have. It's also usually one of the top Europe supercomputers where we crunch a lot of numbers. And now we can move on to this project. So this is actually a project that started, as I said, in collaboration with Material Science EM, and was started together here in this case also with the R&D department of Thermo Fisher, Ivan Lazic and Martin Vieriks, who's been instrumental in establishing this technique and also this kind of equipment for Material Science EM. So why would you venture into STEM, and what is STEM? And I'll give more also as a historical wrap-up on this and try to educate you, because probably this is not the typical kind of cryo-EM imaging. So first of all, why would you bother about different imaging modes? Cryo-EM is incredibly successful, and you probably know this. There were these structures of apoferritin to around about 1.7 something resolution. So this is already a really true atomic resolution because you can identify individual atoms as density blobs, and what else do you need? Of course, we need to use defocus. So this is something we have been used to. This is the common routines in order to generate contrast. And this comes with a trade-off. This comes with the trade-off that the information that we are getting is scrambled up by phase reversals and so on. And this is okay because computationally this can be dealt with. Nevertheless, individual images suffer from modulation in contrast in a predictable but also in a way, a problematic way. So let's be honest, apoferritin is not the typical standard sample that we are working with. In fact, I would say, cryo-EM is really incredibly successful for larger, medium-sized proteins. We heard about that. Now also, in some cases, even inside of cells with ribosomes, we can reach near atomic resolution. But in the end, there is a whole world of other proteins, repertoire for smaller proteins. Obviously, this is something they just don't have sufficient contrast to be able to pick them and to be able to process them and assign their orientations. And I believe also similarly, a lot of the problems that also Grant has just discussed could be helped if we improve the initial contrast and of the original images rather than working with defocused images. And what we have been inspired by is work from the material science community, and this has now been known as tychography. This is probably at the moment the world record in resolution, 0.2 angstroms, and they calculate their resolution slightly differently. It's still utterly high. It's obtained on scandium rare earth scandates, so particular crystals where they measure these kind of small distances. And of course they employ 10,000 times the dose that we are using. So obviously it's not a fair comparison, but still this technology has really enabled this, and this is also way beyond it's close to the diffraction limit here. In this case, this study was limited by the thermal vibrations of the individual atoms. And all this work is pioneered by Dave Muller at Cornell, and he's now collecting a number of prizes, and those of you who've been to the IMC know what I'm talking about. So what is STEM? STEM is actually a large family of methods. And of course, at the center is really that we don't collect an image as a whole, but we identify raster scan positions and then move a probe across the sample. And this, on the one hand, provides us with two-dimensional information about these raster positions, but of course also gives us information about the scattering angles, about the scattering intensity at these individual spots. And then there is a whole lot of other words that you may have encountered in the meantime, and I'll try to put some order to them. So maybe let's start with the more established ones that I think quite a few have probably come across with. So one is the Z contrast. This is the typical high angular dark field imaging, where basically you have a detector that collects one, it's like a circular shaped vox, pixel, if you will, so it's a one integrated signal over that. And then there's bright field STEM, and also you could probably consider now the DPC, it's certainly in the material science world is established, and this is actually what our work what I will show you about. But then there is this whole world of other methods which all are probably can be summed up under this name four-dimensional scanning transmission electron microscopy. There's tychography. Tychography is basically this way of regenerating an image from all these individual scan points and kind of data. And there are various ways to do this. There are direct approaches and iterative algorithms. And within these reconstruction schemes, you can also really account for a lot of the things that are happening actually during the electron beam interaction, such as multiple scattering and so on and so forth. And this is eventually how you end up with 0.2 angstrom in resolution. So, as I said, there are these established methods, and in biology, STEM has been used. This is an example from my PhD time when working with amyloid fibrils, as was so nicely introduced. We used scanning transmission electron microscopy to measure the mass per length, so the mass of our individual objects. If you were looking at particular objects, in this case it was an amyloid fibril, we managed to reconstruct it by pushing helical methods, at the time 8 angstrom resolution of this on the A-beta Alzheimer's amyloid fiber, but we had not really atomic level models available for this. Now this has been resolved. There has been a whole burst of structures in this field coming out because of the detectors, I guess. And so we used alongside here this method and determined the mass per length values. And the way this is done is basically that you have an internal reference, such as tobacco mosaic virus, and there you know the density of those, and the scattering mass directly corresponds proportionally to the mass that you are seeing. And in this way, you can then interpret how many of these particular peptides you will find in there. And so at the time we identified it was 4 to 6. Now the structures have been determined of those types of polymorphs also, and there are 4 models that could be detected here as fully ordered density. Then there is a whole another area of cellular stem, and this is work also cryo electron microscopy. This was work that was first established, I think, by Michael Albaum and Sharon Wolf at the Weizmann Institute. And what they do is basically they don't worry about thinning, they just freeze an entire, here in this case it's a bacterium, but also later on they showed this for cells. So it can be literally up to micrometers in thickness, and interestingly, they still generate contrast. So this is something obviously that would be entirely black in a TEM image. And they combine this with tomography, so they also have really a volume that you can image through that. And you can then analyze things. You're starting to see ribosomal densities. Generally speaking, this is limited in resolution because you do have a large fraction of inelastically scattered electrons that can still be used for image formation, but you're still also blurring your image information. So I would say the resolution is probably here more on the order of 10 nanometers in that scale, although that was never fully determined. So what is now the difference? I told you this is kind of what we use all day long, pretty much in most of the labs, that we generate parallel illumination that then interacts with our sample. Then these scattered electrons, they get captured mainly by the electron lens, and then we have some sort of recording system that is based on our detectors nowadays. And in 4D STEM, or tychography, you would use a convergent beam that has a particular opening angle, and I'll show you this is one of the determinants of the resolution, and you're scanning in these raster positions. And then if you have the right type of detector, you now collect a diffraction pattern for each of these scan positions. And what I will focus on today, and this is something that is still under development, you need very fast detectors if you're scanning large areas and so on. And there's a lot of movement of hybrid detectors becoming available, but this is still ongoing. And so what I will tell you about is a sort of a simplified detector. It's still an annular detector, but it's divided into 4 segments. And this one, gives sort of a very simple image, a 4-segment signal that now you can use. And this is known as this IDPC, so integrated differential phase contrast scanning transmission electron microscopy setup. And there are some nice examples from the literature where they have not just looked at sort of the typical very dose-resistant samples, but also quite dose-sensitive samples. So these are organic, metal organic frameworks of largely organic compounds that here nicely lay down flat on the grid. And if you compare those type of images with the TM, and this is with the IDPC, you already create really highly strong contrast, and these are these large zeolite rings here of organic matter, and they are actually also using on the order of 20 to 100 electrons per angstrom squared. So this is very similar. This is of course not done in ice, but still it kind of gives you a glimpse for how powerful this is, and it can be even in some cases where you start to see individual bonds and so on. So it really has already atomic level resolution. And this is a little bit the setup schematically as you have it. Set up in the microscope, you have your probe. That probe, obviously, then the beam interacts with your specimen. It gets sort of a particular shift by this, based on its own electric field. And then you basically need to capture that signal, and you're always combined basically at the moment with an ADF detector. So in the classical sense, you can look at this in parallel. I'll now show you an example later on. And then you have this 4-segment detector. And now for each of these, you are not really interested in the whole diffraction pattern, but you're looking at this, this deviation, that is induced by this electric field. And now you're looking at these, you're determining simply a center of mass for each, because it allows you the symmetric setup to determine a center of mass. And then you basically plot these differences as a function of your raster scan. And when you integrate this, you obtain back an image. And so this is another example of the work applied. This was done in also with Ivan Lajic, but this was done at the in Beijing at the Academy of Biophysics, also at the Fei Sun's lab. And so they basically looked at plastic embedded sections, cells, and this is what a typical dose series would look like at very low doses. You would barely have any contrast. In the cases of this IDP stem, you already at very low doses obtain significantly improved visual contrast, at least. And then there's also another lab in Oxford from the Material Science World, Angus Kirkland. They have started to do cryotachography in collaboration with Peijun Zhang. And here they look at the rotavirus. This is also ice-embedded. These are ice-embedded structures here that are being looked at. And here they use tachography. And these are the typical kind of curves here obtained by simulations with particular optical parameters. Here's this opening angle as the difference. And you see that basically the image information doesn't oscillate. It does have a profile, but it basically has no zero intercepts or anything alike. And in this study, there was a very recent study coming out in 2023. They've also done some image processing with these images, but at the moment this is limited to 20-ish angstrom. So this is our setup that we have here now at the detector, and it took us quite some time in Jülich to put it up, because it's just not the standard kind of thing that the life science engineer would do. Although in principle it's all even commercially available. So you basically have this HADF ring detector from Fiscioni, and then below that you have this Panther detector, that is this 4-segment detector. And you can run this in parallel to the Falcon and the K3, as in this case you can see. Now, what are the important optical parameters in this setup that eventually also determine your resolution? So I told you have to generate a very small probe. This can be basically a few angstroms across only, and you will need to set up an opening angle, and that opening angle determines your resolution. And in this case, you see the shape of the beam, and as you open up this angle more, you increase your spatial resolution. So this is the sort of maximum theoretical resolution. So here we kind of tuned it into these areas that are at the moment interesting, I think, for the near-atomic regime. And then you should also keep in mind, however, that with this kind of cigar shape of the beam, you also decrease the depth of your focus. So that means the area, the slice, the width that you project through becomes thinner and thinner. And therefore we stuck to something that is still relatively widely open, or not so open, like up to 4 millirads, because then you're still with 200 nanometers pretty safe. But I mean, if you open this up further and further, you will see here in this sketch that at some point in 10 millirads, you're really literally only looking at an optical section of your sample. And of course, this at the moment something maybe for the future, but at the moment not really that so desirable if we understand this in principle. And this is the typical setup. So now we have to bear in mind this is really different to how we regularly image, how we deliver the dose. So we try to make this as compatible as possible in the sense that we create also 4K by 4K raster scans. And then we move them across the sample, and we have a beam dimension of something like 5 angstrom in its half maximum. But we do this also in overlapping steps. And so this step, basically, that every raster position determines now the pixel size in the end. This is what you then obtain with this. But bear in mind that within this delivery, you're also having you have overlapping spots every single time, and therefore also you need to take this into account when you want to really deliver a certain limited amount of dose, of course. Yeah. And so these are the first trials. So we plunge froze tobacco mosaic virus, test specimen that we've been using over time. And now in this setup, you can freeze the specimen, of course, and here create these, almost parallel alignments physically. This happens at a particular resolution, at a particular concentration of the specimen. And then you can also check the signal and look for these layer lines, because this usually gives you a very bright signal in Fourier domain. And here you can nicely compare the ADF image. So this is the one here. It has a quite a nice contrast, but then overall speaking, we don't really have any higher resolution layer lines, starting with the 1/23 all the way to 1/8, for example. But in the case of the IDPC, was already possible to obtain 2 further layer lines from even up to 1/8 angstroms here, visualized very faintly. So that kind of set us on the track. It should be possible to also exploit this for imaging, for high resolution imaging and averaging. So this is an image of vitrified KLH specimen, so keyhole lymphocyanine, also a test specimen of this very large barrel protein. And what you can see here, this is an image taken at this particular opening angle, 2 millirad, and interestingly, now if you, so we need to, it has a very standing out strong contrast. That's one thing that you obviously see. You have the background ice, and then here you have a blob of, contamination, ethane or so. And we need to first, there are often some image ramp that has to do with the lower resolution, with the alignments basically. So we need to also high pass filter these images, and then what is interesting to see that some of the blobs that are usually black actually become transparent, and that's already one indication of the dose delivery and stem. And so then we took some more images of those, and we calculated a 3D reconstruction. Now this is basically relatively straightforward. These images are being picked by regular software. Cryospark or rely on as you wish, and then you just turn off the CTF correction option, because now, as I'll show you in a minute more detail, there is no oscillating CTFs. They already bear the right contrast, and then you calculate these image reconstructions, and this is around about a resolution of 7 to 8 angstroms, where you're starting to see the framework of the alpha helices and so on. So that already was very nice to see that in principle this is possible. And then we looked a bit more detail at these TMV images, and this is nice because we had a whole series of different image data available from our previous work. This was work on the TM where we started to characterize, for example, these K2 detectors, or the Falcon 2 at the time. And this is what a typical image now looks like here, with the tone rings and so on and so forth, and then these layer lines that correspond here to the orientation of these specimens, and then we segment them out and do some processing. But what is interesting now in this IDPC images, you have such a relatively strong streak. This is a result of this limited 4-segment detector. Ideally, you would want to have a complete, multi-segment, not just 4 segments, but this will result in such a streak in the Fourier domain. But apart from that, it doesn't have much effect on the high resolution of the images. And what you see here, there are no tone rings, so you have complete information transfer all the way to high resolution. And then we segment them out, and we compare, for example, here the intensity, the relative intensity of the 1/23 and high resolution layer lines, and this gives us a glimpse for how fast is this information decaying and how good this information transfer at high resolution is really taking place. And in this case, it sort of indicates that the images obtained in 2015 already on the first generation of direct electron detectors have a similar kind of signal between those two, when you look at the intensity of these two layer lines, than the IDPC images. And of course, I need to briefly drink something. Now, of course, when you now calculate 3D reconstructions of this, you can now go more into detail. So this is data from the, what we call here CTEM, so conventional transmission electron microscopy. This is how it's often been termed, and with respect to this scanning transmission electron microscopy. And you can now look at these data that were at the time, this is our highest resolution that goes to around about 2 angstrom. And of course, this is the kind of detailed show up here with the side chains looking out. There was quite a lot of water molecules. And here, this is sort of the smallest opening angle of the TMV reconstruction, and that led to a resolution of 6 angstrom, shows us the alpha helices, so that's a pretty good. And now we opened up this semi-convergence angle further, and now generated reconstruction with the Fourier shell correlation at around about 3.5 angstrom. And now we can look at this density in more detail here. This is an alpha helix, and we see clearly side chain details as expected. And if we compare this to a map at the time from 2015, at a very similar resolution, I guess one can argue that these really don't look very different. And it also goes even to such an extent, if you now look at the negatively charged side chains, and the cracks know this, that these are often tend to be absent or attenuated in all the EM densities because they are the first leaving groups as a result of the radiation damage. And this is equally visible here for this glutamate. That is equally visible in these IDPC stem maps. So I think quantitatively or qualitatively speaking, this looks all very similar. We would like to, of course, know this more quantitatively, and there we did these comparisons, and some people have shown this before in the talk from Saga. The Rosenthal-Henderson plots are now interesting because they give us a sense for how good an imaging system is. And basically what you do is you plot the resolution as a particular number of subsets, and you do obtain a slope. And this slope basically tells you how many images you will need to obtain an X resolution. And if you wanted to go to 1 angstrom, at some point you read really millions or billions of particles, so this is basically unrealistic. But here in all these realistic ranges, so we look at several different graphs here. So one here, the upper one, the green one, is the one from 2019. So this is the highest, the best resolved TMV reconstructions, and there the B factor was determined at 60. And down here we have now this comparison here, the blue and the orange one. This is the IDPC in blue. And it basically has a B factor of 90 as opposed to the CTEM imaging in 2015. So basically, based on this quantitative analysis, the performance of this kind of imaging is between 2015 and 2019 somewhere. So already pretty good, actually, considering that this has not been really exploited. So and there I come to the conclusion in this part of the talk. So we basically know that the highest contrast is here in focus, and we have a complete transfer of information. We have no phase reversals here, and so this we can really take it face value. We showed that this technique can be used to determine near atomic resolution structures, and now we of course want to also push this for smaller specimens and other things that are more difficult to obtain. And also the radiation damage effects are actually quite similar. In scanning transmission, I think there is a lot of discussion and debate to what extent you can actually minimize the impact of radiation because you're delivering the dose in a very different manner to what you would normally do in the TEM. But that I think future studies will need to show to what extent this can be perhaps exploited for the cryo-EM. Yeah. And now in the second part of the talk, I will just show you how we regularly use TEM imaging and to apply to a particular biological system. So this is a system that we have been working on in a collaborative project, the phage shock protein family, PSP genes that are all located on one operon. They occur in bacteria, and they are being upregulated when phages infect bacterial cells. And there is one protein called PSPA. This is basically a membrane protective protein of the system. And there was some idea they interact with the members of this operon, and it's being released in the case of stress, and it sort of seems to have some protective role in how it exactly does it, whether it shields the membrane or else was entirely unknown. It was known that it can sort of also generate punctate type of cells at the poles of these bacteria, and also some fragments of crystal structures had been resolved. And so we purified this protein and imaged it in the electron microscope, and immediately it formed these long helical structures. And we can then use Fourier analysis methods in order to determine the actual symmetry. So this is 10.2 units per turn that make up this helical assembly, and then at 3.6 angstrom resolution we can trace the individual polypeptide backbones and also determine this, the actual conformation of this protein. So what is interesting is basically it's a very elongated shaped protein that forms in a way like brick wall of a cylindrical wall, and then basically makes up such a cylinder. And here this is the structure and also the primary structure. So it has this kind of full alpha helical fold of several different alpha helices that are put in order and called alpha 0, 1, 2, 3, 4, 5. And they make up this elongated molecule with a large obvious hairpin here with a long helical hairpin between alpha 1 and alpha 2. And then this thing, as I said, wraps up here in 10.2 units, and then these 10.2 unit stacks can then be sort of stacked up into this structure. What was interesting when we looked at this structure, this somehow looked familiar, and it turns out that these structures are part of the escort family. So why is that? So the escort family, I'll show you in a minute what they biologically are involved with, but let's stay on the structural side first. So these are structures that are from the protein data bank resolved in Adam Frost's lab. You just mentioned it before his work. And these are also part of the escort family from humans. And then there was another protein that we recently resolved from yeast. And so it became immediately obvious that there is a high similarity in the fold between them. But it's really, at this stage, probably limited to the actual extent of the alpha helices. And when they sort of, the way these helices, these secondary structures are then put together can actually vary quite a bit, as you'll see. And of course here I highlighted them in the particular color corresponding to the particular helix. And this doesn't really look like so related. Now if we look at the quaternary arrangements, there's some interesting observations. Also, they seem to form quite different assemblies. They are all kind of helical extended, that's common to all of them. But large cylindrical structures, smaller ones, they can even co-occur in a copolymer. This is this PSPA molecule that I was just talking to you about, and then this is a helical filament of VPS24, which does not form a cylinder, but it just forms a helix, a ribbon-like structure. And now, although they are very different, what is common to all of these structures is a particular quaternary contact. And this is the contact between alpha helix 1, 2, and alpha 5 here are depicted in this. So all of them have this hairpin structure and this alpha 5 packed against this. And that is interesting because I think it resolves a lot of questions in the field, but interesting in the sense, or astonishing in the sense that this is all consistent, these contacts, while the actual quaternary arrangement is really radically different. In the end, I think this is a common theme of evolution. I think this has also been observed in virus assemblies, where the actual assembly is quite different, while the conservation of the individual proteins is really, really close. And in parallel, there was also some studies that also could identify other family members of this PSPA family, VIP1, as part of this escort family. And in this way, we could independently show that PSPA is part of the escort family. But what does escort do? Escort, common to all is a membrane remodeling machinery, and the escorts are functional, and they had been identified at the membranes of the multivesicular bodies. And what they do is, their paradigm description really is that they're acting inside the cytosol, and then they're pushing membrane away from the cytosol. And this is depicted here, and in some form they assemble and then they push the membrane into the interior of the multivesicular bodies. And this was the first discovery by the Emma lab, in 2001, so quite some time ago. But in the meantime, actually, a lot of cell biology has occurred that involves a lot of the machinery in very different membrane remodeling processes. And this can be, mid-body cytokinesis, membrane repair at the plasma membrane, also nuclear envelope sealing, MVBs I told you about, but also, for example, budding of viruses, because they all share this basic phenomenon that membrane is being pushed away and budded away from a cytosol. So that is really remarkable that such a system can accomplish all these tasks. And now, of course, we would like to better understand how it does that. And we still basically remained to look at this bacterial model system, because the system seems to be a lot less complicated. There's a lot less isoforms involved, and so we can already learn quite a bit. And one of the things when we looked at the data more extensively is that there is a whole variety or diversity of structures. So, for example, we can now use high throughput methods to generate lots of data and then create lots of classification and also see different diameters of these assembly and resolve structures for each of these. So this is an example of 5 structures from that very similar data set that we had obtained earlier, where we just focused on the one assembly. And now we can actually try to understand how they correlate. And if you take these different models now that you generate and you morph them over one another, they really make a suggestive move in a way, although we haven't seen this, but it's obviously a morph, but still it kind of shows you the plasticity of these structures, how probably under different conditions it can undergo these kind of remarkable changes. Well, this is still a single protein in this solution. Now what we of course now want to understand what's the effect on the lipids. And so here we use more heterogeneous systems. We co-incubate them with lipids. And now you look at this. Here EM is a great tool because you see also the diversity of structures that are now in these cryo-EM micrographs. And so on the one hand, you have these large vesicular carpet structures. You have obviously tubes. You saw that before. You have also even like stacked rings. And this is now another type. This is the VIP1 protein, so it's slightly related. But this seems to have really a tremendous effect on these membranes because they're just co-incubated. And of course now you can use classification methods to really cleanly segregate them. And we see here these stacked ring structures. We see these tubes. We see even different types of tubes with very different lattices. Here you see there is more of a step-by-step and parallel, and here is more of a zigzag pattern. And then even if you now segment the densities on the carpets, you also see quite a lot of regularity on those. So we basically addressed them one by one. So we started with these stacked rings, and we could resolve them. These stacked rings are really of cyclical symmetry. They also occur in very different, in a number of different symmetries. So these are all now reconstruction. It's around about from 5 to 7 angstrom in resolution. That's usually good enough to place the molecule. We had built it multiple times. And now what is interesting also, of course, we're starting to see internal density for the lipids. So basically the lipids has sort of internalized, the protein assembly has internalized the lipids now, and you can really follow the path of the lipids. You see this phospholipid bilayer that's sort of squeezed in these helical assemblies. And in these structures, that is even more interesting than that goes from a different symmetry to another one, and therefore also bends these phospholipids more. And we also wanted to address these carpet structures, and there we took a lot more tomograms on this data, and you were starting to see individual ones that are actually not even bound to lipids, but then you see a large different lipid structures, and you see almost like different states of them budding off here from this particular lipid vesicle. And now you can subject that also to the typical segmentation routines, and that makes it maybe easier to read now. So red is now the lipid, phospholipid membrane. The yellow ones are the non-bound lipids, and then the green ones are the ones that are actually bound to the lipid. And now we took more than 100 tomograms, and we now isolated, based on the segmentation, different curvature states. So you can now measure the different membrane curvature, and we basically pick particles according to curvature. And so what we could see is that if we plot, the number of molecules that have been segmented on this membrane, that the denser they are occupied, the more curvature you actually observe, suggesting that when they start to assemble in critical concentrations, they start to make, to form and to remodel this membrane. And then we went on to calculate some of the subtomograms for the highly curved membranes. And although this is not the type of 7 angstrom resolution, it's still on the order of 20-ish, and it allows us basically to fit a model, and here in this case it really is a model that we did not see directly in the sort of more symmetrical assemblies, but we saw them basically sort of, if we extended sort of to a hypothetical larger diameter of something like 20-fold cyclical symmetry, that model could be fitted quite well. And I think in this location you also see how this alpha 5, for example, gives quite a characteristic density that you can actually place on these particular subtomograms as well. And now, looking at these individual segments, and also you can sort them now in retrospectively, of course, but you can sort them maybe in potentially in a stepwise fashion that this may represent also the way these really assemble in solution. And what you can see here is basically this one of the things that you have sort of at the moment in these carpets, kind of a loose association in a sort of a coat, and then once they reach a critical concentration, they kind of debud, pull out the membrane and form these kind of eventually these stacked rings or these rod structures that can then actually also dissociate from this. And what is always interesting, and here you see this in this, that the relative orientation of this alpha 1,2 hairpin really changes throughout these structures. So it's a remarkable structural plasticity of this lattice on this membrane. And interestingly, in this configuration, when they start assembling, it's more like perpendicular to the extension axis of these tubes, and then it starts to become 45 degrees, and then eventually it's really longitudinal along this extension of this tube. Yes, and with that I come to an end. So just in conclusion, we could show that this bacterial phage shock protein system encodes for a protein called PSPA that is part of the canonical Escort fold and family, and so it's really been the first identification of this, because at the beginning it was only thought to be an exclusively eukaryotic process. We find that these structures have a remarkable structural plasticity that we can really nicely address by cryo-EM methods, by the power of these classification methods. And the structural diversity now, together with membrane, also suggests different states of this debugging, of this budding from the membrane. And we think that's a really convenient system to study the bacterial escorts, because the eukaryotic systems are way more complex, and they require several different isoforms, and it gives us a first glimpse of how this in principle may work. Yes, and with that I would like to come to the conclusion. So the story on the escorts in PSPA and VIP1 is done in close collaboration with Dirk Schneider and really driven by Benedict and Esther. We have, and we had in the first part of the talk, I showed you the really great collaboration with the Thermo Fisher R&D department. Ivan and Martin have been instrumental there. And this is our group here. We are also setting up, really venturing into four-dimensional STEM methods really for cryo-EM. And so for those of you who may be interested, we are trying to hire people to be part of this journey. So thank you very much for your attention. I'm happy to take questions.
Okay, many questions already. Thank you so much.
Do you think this is a way for the bacteria to spit out phage? Does it actually try to cinch them off and get rid of them?
We want to address this, more in the physiological situation. But what we think it's doing is basically, so there are these very simple experiments where people drop on, I don't know, alcohol or detergent onto cells, and then they are being upregulated. And so probably the idea, there is some remodeling capability to fill patches, to basically keep the integrity of the membrane. And that would also, for the phage system, probably prevent the phage to attack it and to inject the system. So it will probably work on that level, that it constantly remodels the membrane and therefore makes it more inaccessible to the phage system. That would be my interpretation. But that's at the moment still quite open, because I think it's just phenomenological studies that have been looking at that. Yes.
Okay, very nice talk. So something that always puzzles me is how you can define the handness of filaments. So I understand that once you have like near atomic resolution and at least one alpha helix, you can define if it's right hand or left hand. But in the case of amyloids, once you have like most beta sheets, can you have a comment on other methods for defining the handness? And a second quick question, the VIP1 filaments, so how you sort them out, the different polymorphs?
Yes, so the first question is really an interesting one. I mean, there have been quite a lot of methods actually for in the past where we didn't achieve near atomic resolution so routinely to determine the handedness, and they were based on shadowing, where you can really put it at the rotary angle. But what is important for that is that because you're limiting the resolution in some form, these features that are giving your handedness, and that could be as simple as a crossover twist, they need to be visible at that limited resolution, right? And that if you have a very tightly packed lattice, like a microtubule, you're probably not going to see it unless you have a superb kind of shadowing, and there are some nice examples in the literature also with these freeze etching methods applied. But this is essentially one of the easiest, well, supposedly easiest, but remember the difficult part about this is always the, I think it was at the LMB they used to always say, Tony Krauser always used to say there are only two hands, and in the end you can always be off because you have to do your calibration correctly, and I think this needs to be done as stringently as you're doing your own experiment, because the readout of your detector could have flipped your intensities and so on, and so that's why you really need to use the exact same pipeline of your processing workflow in order to do that. And then there are these other methods that were also done, in the 60s, 70s. John Finch, they did sort of helix tilting, because then you get a particular layer line changes. So that also works, but probably the simplest would be to stick to something like that. And of course, nowadays you could also use tomography. Again, if you have these features at that resolution visible, that you can identify the hand. And then it all translates into the other hands, of your particular alpha helical fold or your side chains and so on. Yes. The first question about the VIP1 classification. So these are all basically methods that are now standardly available in CryoSpark, where you can really collect a large data set and really separate them and work your way through. And I guess large data sizes are the key, but also a convenient way of handling that. And I guess this is where CryoSpark is pretty good at. Yeah.
Very nice work and impressive. I was not aware of these developments very well. But my first job was looking at STEM in a time where there were no field emission guns on 100 kilovolt microscopes, and this was in the 70s. And one of the techniques that I thought was most promising was differential phase contrast by Dekkers and DeLange. They published that somewhere in the 70s. What does the I stand for in your
well, this is so you're absolutely right. The method in itself goes back. So this is from Harald Rose, who has written this up. So and that's, as you said, it's from the 70s. So the I is really this integration step, and I think there are a few minor differences in implementation. The integration step has a very good effect on the noise, so it kind of suppresses the noise even more so. But it's also something that had been actually laid out early on, and it's now it's Thermo Fisher speak.
Yeah, with the new technologies and multiple, I mean, you only have a four quadrants detector at the moment.
That's right, yeah.
But I mean, you would like to have many more spots there, yeah?
Absolutely, yeah. That's the way to go. I agree, yeah.
Here. So the structural plasticity that you show in the final slides remind me to mecanum wheels, which were created to provide plasticity in mobility on trucks, for example. So I'm wondering about not the structural plasticity on the structure itself, but the mobility of the structure in the cell. If that can provide some way of relocalize it.
Right. I mean, I guess that's sort of at the moment something we have actually not addressed, right? What we did, we mix the samples, and then we plant freeze them after an incubation of from 30 minutes or to 2 hours. So we don't really have a time resolution. All we see is an ensemble of different configurations. We do think there is obviously a time correlation, and there are now becoming more methods available to study that also. And it's certainly something very interesting because I think one wants to put this together in as you say, in a mobile way.
Yeah. So I wonder if the energy can be put there, like ATP hydrolysis or phosphorylation or these kind of things to modify the structure. That's it.
I see. Yeah. So that's actually something that we are also heavily investigating with this protein family. So there is data, also other work that has shown that there is an AT or GTPase activity. And we have also measured that. The problem is it's relatively little. It's sort of an order of magnitude or less than the typical ATP hydrolysis that we are used to. So the effects are quite subtle. But we do, I haven't shown you this, the addition of ATP changes the diameter distribution. So it does something to the extent of how these diameters distribute. And so whether it's really hydrolysis or whether it's maybe also very slow binding, I think it's still possible, but it's not the typical kind of Walker ATPase motif that sort of acts very rapidly and that has been characterized in the literature. Yeah, but it's a good point. Yeah.
So, thank you very much. Excellent talk. I have two questions. One is as for like the throughput of data collection when you go for STEM? Because on one side, probably the image takes more time to collect, but the amount of signal in one single particle is much bigger. So how does this compensate for the final particles data set? And the second one is for the amount of data in picotachography. So how people are dealing with that, and what is like the perspective of having something that is reasonable.
Right. So the first question with the data collection speed, I mean, that's clearly, we are not good at there at all at the moment. So these images that I showed you, depending on the exact configuration, some of them took up to 60 seconds. That's all doable if you work with TMV, because you don't need really a lot of images, but if we are looking at the other examples that I showed you, this would be painful, right? So this is something that ultimately depends on this dwell time that you have per raster position, and is eventually a readout question of your detector. So this is something where these new hybrid detectors will play a role, and I think there we are already talking, there's time picks development, so you basically don't read out frames, but you make event detection, and you can then virtually basically make also frames, and this will be extremely fast. So I think there's a hope that we can come down also to seconds of exposures on a similar scale to what we are used to. But we are not there yet. That's totally right, and automation isn't there. So it's all kind of sitting at the microscope, pushing buttons like in the old days. Your second question with regards to if you really do tychography and the amount of data that you are dealing with. So tychography is still pretty diverse in the sense that there is not this one way of reconstructing, right? I showed you this family of algorithms that people are using, and I guess at the moment it's not so clear which one will be best for the cryo applications, because it's this particular application. But eventually, I think, to me, I'm not so scared about this, because experience has shown that technology will improve, and these seem to be in principle possible to address. But I do agree that we are not in no way competitive at this moment in time to how much TM has delivered also in terms of data throughput and so on. Yeah, but I'm not so scared. That's what I'm saying.
Okay, so any additional question? Oh, Bruno.
Sorry, maybe a bit strange, but you showed that the larger the opening angle is, the the smaller is the depth of focus in your sample. Now I'm wondering, in analogy to light microscopy, would it make sense to use this technique to replace tilt tomography, to do tomography in the same way as you do with light microscopes?
Yes, that's certainly an option. Yes, you're absolutely right. So to what extent it will give us really a fully, the full information of 3D is another question. But if you it's clearly known in the material science field that you can obtain 3D information based on a single image, because every time you are rastering, every single time you are seeing a slightly different angle. So there is 3D information on there actually, and you can retrieve this. And so it will be interesting to work this out in such a way that you can perhaps reduce the tilt angles. That already would be of great help, but or even totally get rid of it. Yeah, I can't give you numbers, but it's certainly a direction that is very interesting to pursue. Yes, also scan. Well, then we will have, of course, the dose issue to worry about in cryo. Because we will also probably to some extent damage, the moment it has seen one line, it will be somewhat damaged. And so that all this balance needs to be worked out. What is the cleverest way of doing that? Yeah.
I mean, with tilting you also have several exposures. So
You do. You're totally right, yeah.
You replace the several exposures in tilting by focusing from the top to low.
Absolutely, yeah. Yeah, it's a budget, and I guess it can be worked out. Yeah, can be dealt with. I agree.
Okay, so who fancies a coffee? So let's thank again.
diffusion different action power. So if you change the electron energy from PRGA is the consequence the same compared to traditional bands?
I mean, the idea is similar in the sense, of course, you have this interaction with your sample, and at lower voltages there will be more interaction. And so far, I guess all the trouble was with the lower voltages. It's perfect. It's extremely good for 300 kV, not so good for low. But it turns out for the STEM, we don't need that kind of precision. So it's much less, it's much more tolerant to a poor detector at that resolution. Again, by that voltage, again, how it really does that, and what is the trade-off and so on, I think people have never looked at this. It's more, theoretically, you can make an argument, but nobody has worked this out and done it.
I think that's you need putting it into a cone. So the shape of the cone might be different in terms of different application. And it flies slower, so they might become larger. So if you change energy in high way,
Yeah, so I think it's the, well, I mean, the actual pixel detection is the same as for an image, right? But it's just in the diffraction space. These differences are not so problematic. So you have different, the MTF is not so critical because you have different space. But what the trade-off is, I don't know. It's also something involved.
exciting.
Yeah, just do it. Yeah, exactly. But it's really, but I was just wondering, like, it's not fast, but actually you don't need because it's seamless. So how that's, I mean, that's a Is that yours? Okay, so you use this one for the, what do you use for this?
I used my own.
Ah, you use it.
So I can't tell, but there is a way to connect it. Ask how you know.
Yeah, yeah. Just
She
check.
had it in her head, but I didn't
Yeah,
get it.
there's also one that I think.
Also, around to use it.
No, thank you. I've never had any, never been caused by any virus. Quite lucky. All the 4 vaccines anyway. Shots. Well, I always miss it in the morning. Gandão também. Esse cara liga com.
Será que isso foi? Será que isso foi? Mas isso não fica enfiado aqui. Eu digo fazer uma apresentação bem informal, tá?
Sempre que ficar lendo. Muita coisa pra
Eu só vou decorar aqui. Preciso ler para um ponto que foi o seu faixa de física computacional. E o doutorado física você pode ver
It's you.
Now it's
Good.
So
I can take them.
Okay, so moving on with our program here, it's a great pleasure to have here Rodrigo Portugal. Rodrigo is a dear friend as well. We've been working together already for quite a few years. We have a number of grants together with Vanderlei, Gerson, and Rodrigo was really fundamental to start this whole process with the high-end cryo-EM in the country. Rodrigo did his master's in computational physics and his PhD in biomolecular physics. Then he went to the Imperial College to do a postdoc with Marine Van Heel. So he is the guilty one to bring Marine back to Brazil. and Rodrigo now holds a position at the CNPN, which is the National Center for Research in Energy and Materials, where he coordinates all the cryomicroscopy lab there. And Rodrigo is also the vice president of the Brazilian Society for Microscopy and Microanalysis. He's doing also a wonderful work with our microscopy network in the country. So Rodrigo, the stage is yours. Thank you very much.
Okay, thank you very much, Kevin. Well, thank you to all the organizers, Professor Vanderlei, Hilary, Carol, for being here. It's been a great pleasure to be with all of you. So, So to start my talk, I usually have this like introduction to my talk, and I just realized that I think that's the first time I come to a symposium or congress that every day someone talks about Fernandes Mourão. So to not break the tradition, I will show my slide on Fernandes Mourão. But I'm actually, I used to talk a bit about Fernandes Mourão because of this particular context of Latin America. So, of course, as it was mentioned before, he was very important for cryo-EM, probably very under-mentioned in the history of cryo-EM, not only because of cryo-EM microscopes, the diamond knife, the plunging that he was using for tissues. So there are, I think, lots of good reasons to mention him. But one thing, he was in 1954 the one that funded this Center for Structural Biology in Venezuela, and that was actually the first place that I'm aware someone was doing single particle cryo-EM. So because, of course, not him at that time, but after some time, someone else that we should also mention here in Latin America is Raul Padrón. So Raul Padrón went to the MRC lab, he came back, he started working on the IVIC, so it was called IVIC at the time, and he was like pioneer in single particle cryo-EM. So, and now he's in the US. So, probably some of you will know this picture. I was not there, but you know, like I think Kildare was there, Fernando, there's some people. That was 2007 in Venezuela at CSN meeting, and where cryo-EM was, you know, like being discussed. So, a bit before that, in 2004, so I wasn't aware of things like that at that time, but bit before that, in São Carlos, countryside of São Paulo, there was something that, at least for me, was very significant in my career, and it was having some cachaças with Marine. So Marine was, and that was, if I'm not mistaken, Vanderlei's and Marcia's fault, because Marin was in a congress in South Africa. That's the story I know, and you guys invited him to come to Brazil, so Vanderlei, so that's Vanderlei fault. And he was in Caxambu, and then after Caxambu, he went to São Carlos. And then, just by chance, I was asked to, have him having a dinner and some cachaça. So, and from that moment, Marin decided that he would like to make some schools in Brazil and start doing some things in Brazil. So, and I started coming to the field of cryo-EM. So actually, from 2004, then in 2005, 6, 8, all these dates, we organized cryo-EM schools in Brazil, single particle cryo-EM schools in Brazil. At that time, Marin was at Imperial College, so after finishing my PhD, I went there to join his group. This picture is a picture of one of the schools, so that's the way it goes, like 20 students processing data. And after finishing my postdoc there, which was 2010, I came back to Brazil. I was hired by CNPq, and then we got this really nice project that CNPq in Brazil had for a long time, which was the Science Without Borders, that allowed us to invite people from other countries to come here over a period of 3 years. That was very fruitful for us. So having Marin here over this time, and after he decided to retire, it was the moment to, pay back and invite him to spend some time in Brazil. And since then, he's been spending some time in Brazil. So, a bit about what was like the beginning of cryo-EM in LN Nano. That was the 2100. That's where once we got the first holder, the first cryo holder. That, I have to say, that was a material science center. Okay, so actually, most of the times, biologists are not very welcome in material science centers, to say the least. They talk about contamination all the time. And if you check the benches, our benches are much more cleaner than their benches. So I don't know why they talk about contamination. But nevertheless. So then we started working there as a kind of almost courtesy, because Jefferson was very important. Jefferson Bettini, for the ones that know Jefferson Bettini, he was very important at that time. He was the guy that actually was there like supporting cryo-EM and doing all the behind the scenes job, and also in the microscope. And so we were allowed to use on Fridays and Saturdays the microscope. That was like because other than that, there were users, material science users, so it's really, and Fridays also for maintenance, so most Saturdays. But anyway, so we managed to have a holder, start putting some protein in the eyes. Of course, this image is not very good, but it was the only corner in the hole that had some protein at the beginning. So, and then we start bringing some people to work together. And at that moment, structural biology. So the first group of people that actually started getting interested in work with were like X-ray crystallographers. So we had this group that was like a really like first minute group to join us, which was André Ambrosio and Sandra Dias. André now is at the University of São Paulo. Sandra is Lene Bio in CNPq. They had this nice problem of this protein, which was a glutaminase, GAC, that formed fibers. That was not, at that time, they were working on tetramers, like crystallizing tetramers. But it came out in the electron microscopy that there were fibers. So what we're seeing here is they were cross-linked, but we could see fibers. And actually, then they found in 70 and 71, there were like papers describing that in tissues. But so that was the first time we started working with this GAC. It came to be a nice sample to do some cryo-EM. And that was 2013. And that project evolved. There were lots of drawbacks on their side, also in the microscope side. So there were moments that we didn't work with that. But they eventually took the project again. And we were able to contribute with some cryo-EM. This was the data collection. Actually, Andrea was the person behind all the data processing. So he managed to have classes. It's important to mention, although it's a fiber, it's a helical fiber, but it was processed as single particle analysis. So you pick the particles over the fiber and you process that, and he managed to have this very, very nice and beautiful model. And with that conformation of a fiber, that's directly connected to the activity of the protein. So the protein in that form is not activate. And there is, of course, lots of things to understand behind that. So this was one of very early projects, and it took, it's important to me, it took 10 years to evolve. So things were really like slow and difficult. Another project that is not, we started in 2015, more or less. And at that time, the thing that we have like most easily available was negative staining. And so this group, Richard Garrett, Ana Paula, and Deborah was a master student at that time. They work with septins, which is part of the cytoskeleton of the cell. And the idea, so septins, they are numbered, like you have different proteins, they are numbered, so you have, they form like filaments, and in that case they form like hexamers, could be octamers or tetramers, but in that case hexamers, and there was like one single structure of the hexamer, but actually that structure of the hexamer was based on a crystal structure of a filament. So there was the crystal structure of filament. Of course, based on the filament, you cannot decide the proper order. So there was some electron microscope measurements also done to describe the proper order, which was like described like this: 765, 567. But of course, there was the possibility to be 567, 765, based on the fact that seen as a fiber, you cannot decide which one of the two. So they came up with this idea of putting an MVP on the 5. So although this was very well, I would say, solid in literature, the idea here was actually to test, because there is one thing that is called Kinoshita rule for septins. And actually you can exchange septins of the same group, and they were exchanging the protein 5 for protein 2, the protein 2, which was published by protein 5. So we should have the same thing here. And when we start doing the measurements, we start seeing things like that, okay? And of course, at that time, you just, go back, do everything again 2 or 3 times, and so forth and so on. And we managed, Deborah actually was the one that actually did most of the things. There was one other person before her. But to process the data, there was a lot of different ways that we tried to isolate and to see in classes this MBP, because there is a flexible junction here. So it was a bit challenged to have that, but then we finally got that, the 5 was at the edge, and that was done in a very direct way with a negative staining, but later on in a second work, we managed to then go for cryo, solve the structure, and then it was seen that septin 2 was at the end, and actually septin 2 is the one that is equivalent to 5, so it was all right. And that was actually the structure that is being published by X-ray, and so that was a very interesting work to be involved with. Another thing that was like early stage, but then this one I think we started in 2016, 15 or 16, and that's Chuck Farah and Germanis Groh that are here in audience. So they work with type 4 secretion system that was already mentioned today, but that was the core complex. So core complex here is negative staining, then where we were able to produce some samples, and actually that took a long time because Germain had to work, he can probably mention, but I remember at least like 5 different constructions or so to get it stable. So it was really hard to have the ring well mounted, but then we managed to have. And then in a collaboration with Gabriel Waxman, he went to England and Birkbeck, and using diamond, he managed to have this really nice structure of the core complex. At that time, he was working in Chuck's lab, but I have to mention now his faculty at the Faculty of Ciências Farmacêuticas in Ribeirão Preto. And that group, and what I'm showing now is actually like work they did using our facility. So they have many different systems that they're working with. So this phage was another thing that I think it's nice to mention. So that was German and Chuck too. They managed to solve the structure of this phage, the SHAC 411, with like a normal data collection, but then German realized that in the class sums he could see the tail. So we did like 4 times more the data collection that we had originally, and he was able to process that in C1, so without any symmetry position, and to get a really nice view of the needle. And it's not yet published, but as far as I know, it's in the really final stages of analysis. So in this group, there are other people nowadays working with them, so it's a group that is actually nucleating other groups. And this is another project with Germain, Aline, so different phages they work with. Also Leonardo, that's also here in the audience, is working with also different phages. Leonardo now is faculty at the University of Campinas, just next door of ourselves. So it's really good to have all these people working and working in single particle analysis. Another project, there was a project that was like collaborators from LN Bio in Senepen, was the Mayaro virus, and this project was quite challenging, and that was done by Marine together with Elder, was a student at the time, PhD, no, sorry, he was just hired. And that was a very challenging project, like the virus is a big virus. There was a lot, in the beginning, even like for preparing the sample, the number of particles, there was lots of changes that we have to go through to have that. And at the end of the day, it was able to reconstruct in a very, very good resolution to see. And still, this project is a project that we work with. So there are different projects on virus running on nowadays. And also for us, we don't do yet cryo-electron tomography, but one of the things that we are trying to move is to have some cryo-ET, and that is being done in collaboration with Kildare. So we are kind of exchanging lots of information, single particle and cryo-ET, and this is a project that we are now also trying to do some cryo-ET. Well, so these are the projects in single particle that we've been doing, and some of them we had a direct collaboration, others are just like groups working at the center. And now the next step we want to go, since we are in a material center and we have a very strong area on microfluidics, microdevices, and we have some projects that we really think could benefit from, is to go for time resolved cryo-EM. So for us, it makes all sense on the biological side, also in terms of the infrastructure we have available. And for the ones that are not familiar, just to, well, this is a table of, it's a collection of papers using time-resolved cryo-EMs. And this is the time scale that you can access freezing the sample before some reaction, for instance, is complete. So there are different techniques, there are different systems, but one interesting thing is that even with something that is very direct approach, and I will describe that in the next slide, which is going for microfluidic mixing. You know, we have here microfluidic mixing in the time scale of being able to resolve or to mix things up to 10 milliseconds and then froze it. So if you check, there are lots of things that we can do in that scale. Of course, there are probably some enzyme reactions, some other phenomena that you would need something like this post-delivery, or when you kind of heat the grid with something that you want to mix with your sample, or you heat it up and froze it again. But still, with a process like that, which is a kind of straightforward process, lots of tricks, but straightforward, we can access lots of different biological phenomena. and in a way that I think it's also like does not need much infrastructure. It's something that is not only simpler but also cheaper. Okay, so what's that? In a very simple term, what you do is you have two solutions, you have a channel to mix two solutions, and then you spray that over the grid. And this grid quickly goes into liquid ethane. Okay? So, of course, here is where you're mixing. You can have different geometries. Well, probably there are things that we have also to discuss here at the tip, because you want to spray it, how you want to spray it, what the size of the droplets you want. And that's, in a simple animation, that's what happens. So you have the mixer, there is a spray, and you quickly put that in liquid ethane. So the time of your reaction will be something that goes from here up to here. So this flight time of the tweezers together with this mixing, the size of the channel. Of course, you want to mix that, so there is some time that you have to put the two solutions together, reacting before mixing. So all of that, nowadays there are papers that put that in the 10 milliseconds range. Okay? So here's one example of what is being done. For instance, if you have this reaction here, this is a ribosome, this is a recycling cycle of a ribosome. Okay, you have these two intermediate steps being formed, and then they disappear. So after 1 second, you don't have these species anymore. But if you go for something that is like 100 milliseconds, you will have the presence of them, and you could, sort them out and process the data and have the structure. Okay, so and that's being done, of course, for different proteins now. So what we are doing, we are using more or less the same idea, the same concept of having this device. This is the device we're developing. This is Dani, she's doing a master's in our group. This is Mari, she's a specialist at IM. They are in charge of this project. And this is the device. So you have two channels for reaction. You have here the gas, nitrogen, and here in the tip is where the spray goes off. And if we look into details, actually in terms of geometry, what we can do to try to make a device like better or to evolve something is work on the mixing device or working at this edge of the last needle. Okay, so also this is how we built it. So this is glass, this is PDMS, it's a polymer, so you actually build the architecture you want on using these two components. And here is just a top view of the device. Okay. So what we've been doing, we've been trying to have different designs of this mixing chamber and also of the tips. So we now more or less, we try different ones, and we are trying to follow the solutions here. What we are using for now is like in different samples, we are putting just different colors and follow that, how they mix. One thing that has to be said about microfluidics is essentially laminar flow. So microfluidics does not mix it. Like if you have a microfluidic chamber, a tunnel, the two liquids will not mix. So you have to induce some turbulence. And that's why we have these weird shapes like butterfly or the zigzag shapes, because you need to put some turbulence. But the thing is how you do that in a more efficient way, because you have to do that over a really short size, because you want the reaction to be complete or mixed, fully mixed in a short time. Okay, so this is the setup. It's a very home. So this is a syringe pump, 2 syringe pumps, and then you have the device here, and we can test things here. One thing that is important to be said, like you have to use a syringe pump, because if you use things like peristaltic pump, you see we are putting like 2 colors here, yellow and blue, and that's what you get from peristaltic pump, one after the other. you really need to mix them in this kind of device. So this is as cheap as it can get. It cannot be cheaper than that. Okay, so and that's what happens. So you have this spray at your device. Then here you just put your liquid ethane, and the tweezers has to come through the spray into the liquid ethane. Or you can simply take your device, put inside the VitroBot or another device, of course, as close as possible of the liquid ethane, otherwise the flying time will be also reaction time. We're not interested in that. So when you do that, and then ta-da, everything goes wrong. So lots of contamination, probably because of the air. So you're spraying the water on your grid. Here, what we see here is quite good in terms of, we are seeing the droplets here, okay? So this is one droplet, this is another droplet. But they are with, you can count different artifacts and different problems in this image from like thick ice, crystalline ice, contamination, so forth and so on. So we are now, of course, optimizing that. And I will show later, of course, we have to work also with the grids because the thickness of the ice cannot be regulated only by parameters that you have on your mixing device. You have to go also for self-wicking grids. But before showing that, just let me show like quickly what I was saying about laminar flow. So if we take, so this is an image taken from the channel. So the two liquids, okay, yellow and blue, and then we check them over the channel. It's not easy to see here, but actually if you check the borders, we can see some blue, light blue on the borders. And if you check the intensities of yellow and blue over this line, we will see things in the beginning like that. You have like strong peaks, so it's not mixed, and then it goes for something that is mixed over the channel. So we did that like for different geometries. We test that. This strong peak here is just the edge. So what we're seeing in the beginning, and then what we see after a while. And of course, in different geometries, we'll have like different results because of the efficiency of the geometry. Okay, so this seems to be better than the one before. And we are now moving to develop some geometries that we're discussing internally. This is a one that is kind of helical geometry that of course, it's too long yet, but we want to make it shorter. We couldn't measure it successfully because there are lots of these black areas, so mixing colors here and trying to check the colors is not easy. Well, but this is under development now. So the other thing is when we go for that, the spraying, so you have the droplets and they are thick. They are thick because they are. I mean, so you also have to work with your grids. The solution that we have now, and it's being published and it's on the market, is the self-wicking grids. So Spotton, for instance, Chameleon, for instance, use self-wicking grids. And basically the idea is when the droplet hits the grid has to absorb the droplets, making it like a flat layer of water. That's the idea behind it. So one way of doing that, and that's actually the way that is being used by self-wicking grids, the commercial ones, is to grow nanowires. So you on top of your grid, you grow nanowires, and that's being like in touch with the carbon layer of your grid. So to grow nanowires, there are like physical and chemical methods. Physical methods like you just hit the copper, and we try to go for that. And again, ta-da, disaster. So we try to repeat the method that we have in literature. We didn't succeed. Of course, together with that, we are working on chemical, but the grids were like carbonizing, so there are no nanowires at all, just some rugosity. We also, so that was what was supposed to happen. That was what we got. So we are almost there, as a friend of mine used to say. But then we went to the chemical approach. And it's quite straightforward, and actually now we are trying to mix this with some thermal approach. But then we got the wires, they're quite nice. If we compare them to what we have in literature, so this is what's being published, so we see the wires on the borders, so that's what we are seeing here. So things now are getting better. So we test our device on that, and it's really heterogeneous. So you can get situations like that. So here we have one area of a droplet, another one here. So and you can have really thick ice like that. Okay, but at least it seemed to be cleaner. But also you can get areas like that. What we're seeing here is contamination. There is no protein in the sample. But the ice seems to be, at least there are good areas of ice that you can get from that. So that's the point that we are now. Daniela now, this last 2 weeks, she's working with doing some protein test on that. But actually there is at least one work done with apoferritin, where they compared the structure of apoferritin in a spraying device with the structure, the deposit structure, and it's exactly the same. So there's no problem on spraying proteins on grids. They will be apparently preserved. Why is that important to us? So I will show another project. This is a project from a researcher in Senepen, Mario Murakami, and that was done with us. We collaborated in this project, and it was fun because he is an X-ray crystallographer, so he was working with this protein for time, and he was not able the protein was not able to crystallize. So he came with this idea to try to do cryo-EM, but he was very, very clear. Like, I'm interested in a structure of high resolution. We have the active site, we have to see the active site. So, I mean, don't even bother if you cannot go for high resolution. But it's interesting because they've been working for so much time with this protein. When they came with the sample, we did a quick test in the morning, and in the afternoon we were doing data collection in the cryos because the protein was kind of like a really, really perfect protein. And so we managed to have, and actually what we're seeing here is 3 different proteins. They are part of this chain of metabolism for high mannose N-glycans in Bifidobacterium longo. And it's important because actually this is the most abundant source of, like carbon. So this breakdown metabolism is really important for the bacteria. And of course there is one of them is a mutant, but there are 3 different proteins in this metabolism pathway. And why, how is it related? So the next step here, they did some molecular dynamics with these structures to think about the mechanism for the action of the enzyme. And what we want now is to use that as a template or as one of the projects that we are going to try in time-resolved cryo-EM. Of course, it really depends on the time scale of the process that are going there that we don't know yet. The other thing, that is a project that I worked a time ago that we want to pursue is this swapping of DNA on RNA polymerase. So this is a long story. This is a project that we started with Daniel Guerra. So Daniel Guerra from Universidade Peruana Caetano Heredia, he was working this project, and a long time ago we met and we decided to try to do some, at that time, negative staining, and later on cryo-EM. But the idea here is they know by AFM that if they measure the size of a piece of DNA when the DNA is together with RNA polymerase, there is a dislocation in the size of the DNA. So if you check the DNA, it does not go like that for the RNA polymerase. So the model is that it wraps around somehow, and this wrapping is important to form the bubble and to start the transcription process. So that's what we tried to do at that time. We did some negative staining. We never had a convincing model. Then we went to cryo. The student that was working with us at that time, from a few years now, is working in Berkeley, and actually he tried to do that there as well. And the conclusions are like there are so many different states, and it seems that this state is not really well populated, so it's something that is hard to go. So the idea is to use that as also a system that we are interested in seeing with time-resolved cryo-EM, if we can access time scales of the initial formation of the complex and the bubble. So let's see. So that's the overall idea I would like to go through. I would just like to mention a bit more of our center. So this is the National Center for Research in Energy and Materials. That's the center. We have here the synchrotron, the new generation, the new synchrotron. We have biorenewables laboratory, bioscience. This is the old synchrotron. Actually, it does not look like, but this building fits inside this ring. So it's really different scale. And here we have LN Nano, and all the microscopes are here. So this area of LN Nano, like half of this building, is actually electron microscopy. And again, it's material science and life science. So we have 7 TEMs, 4 for material science, well, 3 actually. 1 is kind of hybrid, and 3 for life science, plus SEMs and dual beams. This is the way we work. So we have like open facilities. We operate open facilities. We have in-house research, support for innovation, education, and dissemination. And that's the building. So that's the top view of that building and how we have. So that's mostly what we have, 1,400 Arctica and Krios, two labs for sample preparation. That's how we grew. So we start with a 1,400, 2015, then F200C, Talos, Arctica, and Krios. This one is now receiving microelectron diffraction. This one we have picotachography actually. So this is one of 4D STEM actually, 4D STEM. Also IDPC we have in this one, but sorry, not the Krios. We have a similar one at TEMIS for material science that we have that. But as material science equipment, it does not have a good cryo protection for the samples, so we cannot do the fancy things customers showing us. So, but well, maybe in the future. Also, all the infrastructure for sample preparation and data processing. So, of course, there are also courses offered. This is something that we are starting now, which is like visiting centers that are starting to do cryo-EM or negative samples, negative staining samples for using larger facilities. So we train people on their sites, which is being shown like really, really powerful to improve the quality of the samples of the projects that we receive. And this is something that Kilder mentioned that we've been working a lot in different projects. This is something that we've been discussing a lot, the formation of a network for cryo-EM in Brazil. So this is something that we are more and more trying to put to work. We'll not take long on that. So there are lots of centers that we are discussing, formation of networks. And that's all the people that I would like to thank, people that allow us to collaborate in their projects. And with that, we are really, really involved more and more with really exciting and new things. And this, okay, so this is our group. Not a big group, but here is like Marine and I, Antonio, Mariana, and Beatriz are cryo-EM specialists. Dani is a technician in the lab. We are also a master's student. Skype and Sayan are postdocs, and Mateus is a PhD student. And with that, I would like to thank you all. And thank you.
Adan?
Hi, Rodrigo. So I have a question related to the mixing in the micro devices. I wonder if you have thought in using sound for mixing.
Yes, yes. Ultrasound is one of the ways for doing that. So one of the things that we think is maybe we should have on the bottom of the device some ultrasound to make it faster, also for producing smaller droplets. So you can think about ultrasound at least in two different moments of that. Yeah, but that's important strategy too. Okay.
Questions? Farina?
Thank you, Rodrigo, for this wonderful presentation. I don't know if I understood the objective of the freezing, fast freezing in the range of microseconds.
Yeah.
I thought about the work of Ahmed Zevail, which created femtochemistry, which is the dimension of changes in molecules, changes
Yes.
in conformation of molecules. I thought you were trying to go into this subject.
Yeah, but
Am I right? Because even in his book from 2009 or 2014, he mentions that he can get attoseconds, which is the Nobel Prize in Physics now. So probably in the near future we will have some 4D TM with attoseconds.
Yeah.
But what can you get previously to do experiments in this range of milliseconds?
Well,
for molecules conformation.
Yeah, what we have now is situations like I showed here for the ribosome, a reaction of something. In this case, the dissociation process of the ribosome, okay? And that goes through different steps, that goes through different intermediate conformations of the macromolecules. That could be different conformations, or it could be different complexes. And okay. So I think that's what we, at least with this technology of mixing, that's what we can achieve. We will not get it like too fast to follow like fast reactions. But there are other technologies being discussed that aim to faster reactions. Okay, but then it's like using, for instance, lasers to locally heat your grid and mix something. So. Sorry, let me see if I understand what you're saying.
And the needle-like crystals that you showed that you try to grow, going inside the hole of the grids, should it be for changing more fast temperature of the sample or for other reason?
Okay, let me explain it again. Let me go there. So the needle-like structures you're saying is probably self-wicking grids here. That's what I mean. This. Oh.
There's a cat there.
Okay. So that's what you mean, right? So this is actually on the grid. This is to create a sort of sponge on the grid. So when I thought it was related to the heat capacity of the No. It's to dry the grid. It's to that's the principle of the self-wicking grid. You have this sponge of No. It's just to absorb the excess of water. Okay.
So John?
Wonderful. This is more a suggestion than a question. We did time-resolved membrane fusion by putting membranes, suspension liposomes on a grid, a thin film, and then passing them through a mist of calcium as on their way to the freezing. And I just would suggest to you that you put solution A or solution 1 on the grid, thin film, and pass it through a mist of solution 2.
And then
as
plunge.
It's frozen.
Yeah.
Yes, because the advantage is you have a droplet then, and the droplet is diffusing. So you have all times presented by how far that droplet is diffused. In the center, it's the longest time.
Thank you. How is the ice thickness while you start with the solution on the grid?
Well, that of course you determine. You decide, and you know how to make the first film very, very thin, like you would normally for plunge freezing. And then this mist are 10 to 25 micron droplets.
Okay, thank you.
A follow-up question, Rodrigo. How do you calibrate the system? How do you measure time?
That's what we are trying to do now. So the moment we are measuring the droplets to have them small as possible and checking the thickness of the ice. So, but the further calibration is actually something that we want to.
I mean, time calibration. How do you
Yeah.
know if it's 10 milliseconds or?
We don't have it yet. We don't have it yet. What we know is like checking with the callers. We know that if it's mixed or not and when in the channel. So we can calculate because we know the diffusion, we know the flow. But we are really not at the moment that we are concerned with that. The other thing is, I know we can use, for instance, pH variations, so we can go for that. But for now, if we don't have like a setup of parameters that we can at least see in the microscope, I mean, it's pointless.
Any further questions? So then we thank you again.
Thank you very much.